10:59.
So nice to see a all women's group.
दिक्कत आएगी
तो फिर यार कैसे करोगे?
इधर रख लो ना, इधर रखो
पलट देना फिर मेरा उधर वो उठाना पड़ेगा
आराम से पॉज के साथ।
हाँ पॉज के साथ वो मैंने सारे पॉज
स्टार्ट करें।
स्टार्ट
कैमरा चालू है अब करो।
Good morning, everyone. I am Dr. Alpna Johari.
I'm audible.
Am I audible?
Yes, ma'am, you are.
Yes, you carry on. Okay. Good morning, everyone. I am Dr. Alpna Johari. Respected Dr. Anita Kothari, ma'am, Principal, JDB Government Girls College, Kota, patron of this conference. Respected Dr. Sanjay Bhargav, sir, who is Assistant Director, Kota, Directorate, College Education, Rajasthan, Chief Advisor, Dean, and Member Secretary, DLQAC, Dr. Satish Saraswat, sir, and Chief Advisor, Senior Faculty, Dr. Madhu Khanna, ma'am, our convener of this conference, and HOD, Department of Botany, Dr. Pratima Srivastava, ma'am. On behalf of JDB Government Girls College, Kota, I welcome one and all in this international e-conference on current trends in research and biodiversity, environment and plant resources. जैसा कि हर कार्यक्रम से पूर्व सरस्वती माँ की वंदना की जाती है, मैं आप सभी को सरस्वती वंदना के लिए आमंत्रित करती हूँ।
ये करना है, यस ये करना है, शेयर करना है, शेयर अब आवाज तेज करो।
मेरी बात आपको भी नंबर ऊपर ही आते आपसे तो कहते ही नहीं उन्होंने कहा कि आपने ये दोबारा पहले तो 3 कोर्स तो आपने चालू कर दी, उसकी परमिशन वो दे दी थी तो 3 के साथ
Welcome back. This is 2 days international e-conference and Department of Botany, JDB Government Girls College, Kota welcomes you all. First of all, I would like to invite our respected principal, Dr. Anita Kothari ma'am, for her welcome speech. Now, I welcome our principal ma'am.
Good morning.
Ma'am, mask.
Good morning. Am I audible? It gives me immense pleasure to welcome you all in this 2 days international e-conference on current trends in research in biodiversity, environment, and plant resources, organized by the Department of Botany, JDB Government Girls College, Kota. I feel greatly privileged to welcome our distinguished guests and eminent speakers, Dr. N.K. Dubey, FNAS Science, FNAAS, Professor of Botany, Banaras Hindu University, Varanasi, Dr. Padmanabh Dwivedi, Professor and ex-head, Department of Plant Physiology, Institute of Agricultural Science, Banaras Hindu University, Varanasi, Dr. Somanath Datta, Professor Botany, Dean Faculty of Science, University of Rajasthan, Jaipur, and Dr. Lalit Batra, Postdoctoral Associate, Center for Predictive Medicine, University of Louisville, USA. I would like to extend my warm welcome to Dr. Sanjay Bhargav, Assistant Director, Kota, and Dr. Satish Saraswat, Convener IQAC, Senior Faculty Member, Mrs. Madhu Khanna, and Head Botany Department, Dr. Pratima Srivastav, and all the faculty members, participants, and research scholars. This conference on current trends in research in biodiversity, environment, and plant resource is a very topical and burning issue. The three terms, biodiversity, environment, and plant resources, are interconnected. Biological diversity is often understood in terms of the wide variety of plants, animals, and microorganisms, but it also includes genetic differences within each species and the variety of ecosystems, namely lakes, forests, deserts, agricultural landscapes that host multiple kinds of interactions among their members, namely humans, plants, animals. Biological diversity resources are the pillars upon which we build civilization. But biodiversity is being lost due to the loss of habitat, overexploitation of resources, climate changes, pollution, invasive exotic species, diseases, hunting, etc. Loss of biodiversity threatens all, including our health. It has been proven that biodiversity loss could expand zoonosis, diseases transmitted from animals to humans, while on the other hand, if we keep biodiversity intact, it offers excellent tools to fight against pandemics like those caused by coronavirus. While there is a growing recognition that biological diversity is a global asset of tremendous value to future generation, but at the same time, the number of species is being significantly reduced by certain human activities. Given the importance of public education and awareness about this issue, the UN decided to celebrate the International Day for Biological Diversity annually. The theme of this year 2020 is our solutions are in nature. As the global community is called to re-examine our relationships to the natural world, one thing is certain: despite all our technological advances, we are completely dependent on healthy and vibrant ecosystem for our water, food, medicines, clothes, fuel, shelter, and energy, just to name a few. So the emphasis this year, the year 2020 emphasizes hope, solidarity, and the importance of working together at all levels to build a future of life in harmony with nature. This two-day conference, the main purpose of this two-day conference is to throw light on various aspects and issues related to biodiversity, environment, and plant resources, and acquaint researchers about current trends and open up a whole vista for them to work in this arena so that we can further claim the necessity of biodiversity by considering our degree of dependency on the environment. I am sure by the end of this conference, by your active participation and deliberation, the desired purpose of this conference shall be accomplished. All my best wishes for the success of this conference. Thanks.
Thank you, Principal Ma'am, for your motivational speech. And now over to Mrs. Anita Malav.
Good morning, one and all. I would like to welcome Dr. Am I audible?
Yes.
Am I audible?
Yes.
Please. Good morning, one and all. I would like to welcome Dr. Sanjay Bhargav on behalf of Principal JDB College and Department of Botany. Let me introduce to you. Dr. Bhargav is currently working as Regional Assistant Director, College Education, Kota Region. He has a vast teaching experience of more than 35 years. He has 8 research papers in internationally reviewed journals, 9 papers in conferences and symposium, and 3 books to his credit. Dr. Bhargav has also worked as convener BOS in Physics, University of Kota, co-convener Grievance Committee, University of Kota, Principal Government Girls College, Jhalawad. We are honored to have Bhargav sir amongst us. He accepted our request readily. We appreciate your humbleness always. Welcome, Dr. Sanjay Bhargav sir. Over to you now.
Ma'am, am I audible? मेरा आवाज आ रहा है मैम?
Yes, sir.
In the two-day international e-conference organized by Department of Botany, JDB Government Girls College, Kota, present respected principal of the college, Dr. Anita Kothari ma'am, the eminent speakers of the conference, Professor N.K. Dubey, Dr. Lalita Batra, Professor Somna Datta, and Dr. Padmanabh Dwivedi. Convener of the program, Dr. Pratima Srivastav, DLQAC Secretary, Dr. Satish Saraswat, members of the organizing team, and participants of the conference. It has been a sheer delight to be a part of 2 continuous day international conference, and both related to more or less identical topics of biodiversity and environment. Being a student of physics, it just gives me an immense satisfaction to be the part of biodiversity, environment, and plant resources. These words, which form a very relevant and most important topics of deliberation in the modern times. For me, the knowledge of the words may be superficial, but as a layman, let me say something in front of you all. Biodiversity refers to the variety of plants and animals found on Earth. It measures the variations at ecosystem, species, and genetic level. Biodiversity is the variation in living forms and is measured by the ways which include the number of species, the functional variety of species, evenness of species distribution, or the genetic diversity. The biodiversity science investigates the levels of biodiversity, its functional effects, and how and why it changes over time. Biodiversity is being lost due to loss of habitat, overexploitation of resources, climatic changes, pollution, invasive exotic species, diseases, hunting, etc. One of the main problems of biodiversity research may be the vastness and the nature of the data that have been or will be recorded in future. Biodiversity data encompass a multitude of structures, from DNA sequences to data on physiology, morphology, ethology, population, community ecology, and habitats. The scientists doing research work on both the scientific and the social scientific dimensions of the functioning of ecological and biogeographical systems, with principal interests in macroecology, island biogeography, the diversity theory, ecosystem dynamics, conservation governance, biodiversity and the climate adaptation, and conservation and traditional ecological knowledge. In the modern times, the biodiversity research is very important and has a high priority, just because the demographic growth of human population and the exploitation of the natural resources have led to a progressive erosion of biological diversity. The consequences of this erosion can be seen by the increasing extinction rate of the species, the fragmentation of population, or reduction of genetic diversity. The scenario is likely to become even more worrying with the expected global change. The growing awareness that biodiversity is a precious global asset to the present and the future generations, and that the survival of the species and the integrity of habitats and ecosystems are at serious risk, has increased significantly the importance of biodiversity related research. The acknowledgement of the importance of research on biodiversity and the development of new, high sophisticated methods for measuring natural variation at the genetic level has led to a considerable impact of evolutionary biology on the biological sciences. Today, evolutionary biology influences significantly the hypothesis and the research programs from the other areas of natural sciences. It has been found that a fundamental knowledge of the evolutionary processes underlying biodiversity and its variation is necessary to understand many biological systems. In the biodiversity and ecosystem research, people study the structure, function, and change of the terrestrial ecosystems by using plants, vegetation, and soil as integrative key features of landscape ecology. Processes of global change, especially that of man-made climate and the land use changes, as well as the changes in the geochemical cycles, are studied on local and regional scales with regard to their importance for conservation of biodiversity and sustainable use of natural resources. The environmentalists continuously devote time to look at the continuous environmental as well as the ecological trends which face our global society. As far as my understanding goes, in 2020, some major environmental and ecological trends that people will be working for may be the increase in renewable energy sources. For last many years, there has been a debate between the advocates for renewable energy sources such as wind and solar and the traditional energy sources as coal. In last few years, the use of renewable energy resources has been gaining popularity. In 2019, the renewable energy sources surpassed the coal as the source of electrical energy in the developed nation like US. Another thing is reduction of energy consumption. The demand for energy that is projected in 2020 is, as far as 2019 is concerned, shows a decrease. As individuals have become more environmentally conscious, they look for the ways to minimize the use of energy. This may involve significant changes in the lifestyle so that less energy is utilized. Another way of reducing the consumption may be to switch over to the alternative renewable sources. Another important word in this area would be the regenerative agriculture, which is a method which prioritizes soil health, land management, and biodiversity during the cultivation and harvesting of food products. With regenerative agriculture, deliberate practices are used which minimizes the loss of necessary soil nutrients and minerals, thereby preserving the life of the soil. Another key factor may be the air travel and the climate change. What I read, air travel is a significant source of greenhouse gas emissions which are harmful to the environment. It contributes about 2.5% of the global carbon emissions. Due to this grim statistic, the research is underway to make aviation more energy efficient as well as eco-friendly. Efforts have been made to the biofuels to provide biofuels as an alternative fuel source. Also, research is going on for the development of electrical and solar powered planes. The word biodiversity, environment, plant resources, they all are closely knit words. And what I hope during the 2-day conference, we expect all the important, eminent keynote speakers to rather provide the participants some new directions of work and research, which may go a long way in planning their research career. I wish all success to the seminar and fruitful results and deliberations. I, from bottom of my heart, is thankful to the organizers of the e-seminar who have provided me an opportunity to just share my ideas with you all. Thank you. Thank you very much.
Thank you, Alpna. Respected Principal Madam, JDB Government Girls College, Kota, Dr. Anita Kothari, Dr. Sanjay Bhargav, Assistant Director, Directorate Region, Coordinator IQAC, Dr. Satish Saraswat Sir, Mrs. Madhu Khanna, Senior Faculty Member, and all our keynote speakers, Dr. N.K. Dubey. I need to mention that he has been selected among the eminent 19 scientists by the Stanford University. It's an honor that you have joined us. Dr. Padmanabh Dwivedi, Dr. Somnath Datta, and Dr. Lalit Batra, our international speaker. I welcome you all to the 2 days international conference on CTRBEPR 2020. It is just an honor to hear Dr. Anita Kothari Madam and Dr. Sanjay Bhargav Sir. Though these people are not from botany background, they have really enlightened us on the subject. Now the thematic areas of our conference. I mentioned here the thematic areas of our conference. Sanjay sir has rightly mentioned as three terms: environment, biodiversity, and plant resources utilization are interrelated. We all know biodiversity is collapsing worldwide, environment is degrading, and plant resources are depleting. The question arises, who is responsible? Of course, we the humans who have destroyed everything for our needs and greeds. But this doesn't mean that we should stop development, growth, and other activities just in order to save our environment. Now, the basic question is, which is really the purpose of our conference, is to generate awareness among the masses, to bring good work in the terms of good researches under the thematic areas which we have taken under. During these 2 days, there will be oral presentations and our keynote speakers will highlight on biodiversity, plant resource utilization, pollution researches, environmental chemistry, sustainable development, and please आप लोग अपना प्लीज म्यूट कर लीजिए. And I would like to end on a high note because much has been spoken earlier by our esteemed, respected speakers. I would like to end on high note that definitely you all will be going to be benefited, and we only need to introspect about all these issues. In the end, we will have oral and poster presentations by our researchers, scholars, and really, I'm thankful to all the participants for their overwhelming responses we have received, not taking much of your time because Dr. Batra is online and he is our international speaker. So I end up here. Only the thing which I want to say, this is the right time to wake up. Otherwise, our future generations will be sleeping forever. This we are seeing in the current scenario. Over to you, Dr. Alpna Johari.
Thank you. Thank you, Dr. Pratima. Due to unavoidable circumstances, there is slight change in schedule. Dr. N.K. Dubey will deliver his lecture tomorrow at 11:30 a.m. In place of Dr. Dubey, our first keynote speaker is Dr. Lalit Batra from USA. We are very thankful to Dr. Lalit Batra who has connected to us on a very short notice and at very odd hours. It's my pleasure to welcome Dr. Lalit Batra from Louisville, Kentucky, USA. Dr. Lalit Batra is postdoctoral research associate from Center of Predictive Medicine, University of Louisville, USA. I'm very proud to say that he is an Indian researcher doing his work at international level on very important subjects like cancer and immunology. He has been working in Louisville since 2050 after qualifying GRF, NET, and also got a scholarship from PBG India. He has teaching experience also in India and he is recipient of many awards at national and international level. Like Young Investigator Award and 2 awards in immunology and many fellowships. He has many international publications and has presented papers in many national and international conferences. He has done many specialized training courses on environment, health, and safety by University of Louisville. He is member of 4 national and international bodies. His achievement speaks volumes about how Indians have established themselves in advanced researches and brought laurels globally. Students, ये हमारे लिए मोटिवेशन है कि हम किस तरह ग्लोबली भी काम कर सकते हैं. Think locally and act globally. Today, he will be sharing with us his expert talk on GRFT, a broad spectrum antiviral protein. Now, I welcome Dr. Lalit Batra. Welcome, sir.
आवाज नहीं आ रही.
Hello everyone. Thank you, Alpna, for your words. Thank you very much. Let me start with the screen share. It says screen host disabled participant screen sharing. Actually, I want to share my screen for the PowerPoint presentation, but it said host disabled participant screen sharing. So can you see my screen? Can you see my screen?
Yes, sir. Carry on. Carry on, sir.
Thank you very much. Thank you. Thank you again, Alpna ma'am, for your words, for my introduction. Let me introduce myself to everybody again. So myself, Lalit Batra, I did my master's in biotechnology from BHU Varanasi, and then I did my PhD from DRD Gwalior, that is the Defense Research and Development Establishment. There I worked on the plague vaccine research. Then in 2015, I came here in United States for postdoctoral associate position. And here I'm working in a lot of projects like cancer, diabetes, immunology, and recently on COVID-19. So let me start with my first project. So my first project was on the cancer immunoprevention with SA4-1BB ligand. So we SA4-1BB ligand molecule is a important regulator for the 4-1BB ligand pathway. This 4-1BB ligand is a important regulator for the Th1 immune response. It activates a lot of innate immune and activate the other component of the system. So here we generated a novel form of the 4-1BB ligand. It's a SA4-1BB ligand. So we conjugated the streptavidin with this 4-1BB ligand. So with this, it forms a good compliance and it has a tendency to form oligomer and tetramer, and it can be, you can load any antigen on that and it will be delivered to the targeted antigen site. So initially in our lab, before they work on this adjuvant. This is SA4-1BB ligand adjuvant, and they use for the treatment for the HPV and other cancer antigen, and they treat the mice with this as an adjuvant. Later on, when I came, I started working with this adjuvant, and then I observed that with this treatment alone with this adjuvant, there is no need of any cancer antigen. So we can give it to the mice, and the mice, when we give this immunize with this SA4-1BB ligand, and then immunize the mice, and then give the tumor, the mice they were tumor free. So first of all, I give the first of all, immunize the mice with survival cancer model.
Participants, please mute yourself. Again requesting, please mute yourself. Participants.
वो आ गया ना सोडियम वाला तब से मतलब अलग से हाँ इसके बदले melanoma and the 41 breast cancer cell line. So the most importantly, this immunopreventive efficacy of 4-1BB ligand, it starts from the 2 weeks and it is there until the 14 weeks. As you can see from the graph, so the efficacy started from the 2 weeks, 4 weeks, 8 weeks, and it remained until the 14 weeks. And later on, then we characterize why we did this as a prevention agent. So this is important for the patients that have the hereditary to some mutation. They have P53 mutation or the preneoplastic lesions, or they are mute, or the person working in the smoke cigarette industry, which are more vulnerable to cancer. So this molecule can be given to them, and they can be cancer free. So, and later on we decipher the mechanism. Later on we decipher the mechanism behind this prevention, and we found that CD4, CD44, NK cells, they are playing role, and the CD4 and a lot of CD4, CD44 cells are interferon positive, NK cells are interferon positive, and NKT cells are interferon positive, as you can see from the significant bars. So this research was published in the Cancer Research in 2019, and part of this research has been in communication with the Nature Medicine journal. So this is my first project on cancer. Now my other project was on the diabetes. So in the diabetes, we are using on the islet transplantation model. So the people, there is a new approach. The people who are type 1 diabetic, they can be given to the islet, so that the beta cells, they are functional. So they can be treated with this islet transplantation. But this islet transplantation has some problem, because the people has to be on the donated islets, but there is a like allogenic response. If somebody else can, he will give you its tissues or organ, there will be a lot of allogenic response, and you have to be survived on a lot of immunosuppressive drug. So we want to cut off this immunosuppressive drug regimen. So here I use the immunosuppressive molecule, that is a PDL1. So similarly, we ligated with the streptavidin to form a oligomer and tetramer, and then when you have like biodiversity cells like islets or spherocyte, you can very well add on to them, and they are really nicely decorated. So these are the bindings that are targeted with the SAPDL1 molecules. So this mechanism is our experimental, how we perform the experiment. So they are the P6 mice. First we give them streptozotocin. It makes the mice diabetic. That is, it kills the beta cells of islets. And then these mice are diabetic within like 4 days. Here we have isolated the islets from the other mice like bulb C. So they are variety are allogenic. B6 and biopsy we are allogenic mice. So we harvested the islet from this and decorated the PD-L1 protein onto them and see how the graft are surviving. So to our surprise, when we give the SAPD-L1 only, the mice survive in expect to control up to 40 days. But this treatment was not effective. But when we give the SAPD-L1 only, we use pressure rapamycin, not for a long time like for the generation we have to give for like short course of 15 days. The mice they were diabetic free until the end of experiment, that is the 100 days. So with this notation, this molecule is under the it is non-human under the non-human primates. Now this experiment was on mice. Now this regimen we are applying for the non-human primates like monkey, so that a short treatment of rapamycin suppress the allogenic response and we can target the diabetic, we can overcome the diabetes. For the mechanism, we see that with this treatment we are seeing a lot of regulatory cytokines like FOXP3, TGF-beta, and a lot of anti-inflammatory cytokines like IL-10, IL-4. On the other hand, we see a increase in the and decrease in the pro-inflammatory cytokines. In the red bars, you can see the red dots, a decrease in the pro-inflammatory cytokines like interferon gamma, TNF-alpha, and IL-1 beta. When we, at the end of experiment, at 100 days, we harvested the kidney from these mice, and we see a lot of CD4 cells with FOXP3 positive. That means these cells are the regulatory cells. So they are playing the roles, CD4 FOXP3 cells for their mechanism. So this research has been published this year in Journal of Immunology, Science Advances, American Journal of Transplantation, in two journals. So this is like a separate part of what we publish in different journals. So the Science Advances is held for the non-human primates. We publish that for non-human primates. So this is my second project. My third project was on the infectious disease. So in India, I work on the plague vaccine. Here I got a chance again to work on the plague vaccine. As I told you earlier, this SA4-1B ligand molecule, I work with SA4-1B ligand molecule for the plague vaccine. Plague, everybody knows that plague is caused by first Yersinia pestis. Like we are seeing this COVID-19 pandemic, there was like a long back ago, there was a pandemic for the Y pestis, it's called plague. It killed a lot of millions of population, way back in 1500 or something like that. So in India, the people in the DRD, they are working for the development of vaccine against this plague, deadly bacteria plague. Here, the people, they are also working on the development of vaccine against plague. So here, for the plague vaccine, what we did, we have the F1 and B antigen of Y pestis, and we have given the mice to the F1B with SF1B ligand protein. So this is a like short brief experiment. So here we generated 3 groups. So in which we give the F1B with L-hydrogel and boost it again with this regimen, and then we give the F1B with L-hydrogel and then boost it with F1B with 4-B ligand. In third group, we give the F1B with L-hydrogel and 4-B ligand, F1B with L-hydrogel and we give 4-B ligand. So as you can see, the third group has maximum amount of interferon gamma 0.186 from the CD44 cells and 1.53 as compared to other groups, 1.53% of interferon gamma coming from CD8, CD44 cells. So as you can see in the form of absolute cell number, we see a lot of a very significant amount of interferon from CD4 interferon from the last group, that is F1B with the hydrogel and 4-B ligand with the same boosted dose. Similarly, we see the CD8 interferon gamma T cells. Here we see a lot of IgG2C coming from the last group, that is hydrogel plus 4-B ligand. So when we give the mice the challenge with like 10, 50 to the mice, to the immunized mice, we see a like reduction in the survival, but we didn't achieve the complete protection. Then later on, we are standardizing the doses of the vaccine, and we are getting the complete protection. This research has been published in Vaccine in 2019, and the part of the work is that we see the complete protection is again communicated in the vaccine journal as a part 2. So as of my interest in my PhD and in the postdoc for the infectious disease research, I came into the play for COVID-19. So I started working for the COVID-19 therapeutics. So for the COVID-19, we are working for the therapeutics for COVID-19. We are utilizing a lot of antiviral and other vaccine approach for the prevention for the COVID-19. So here we have today novel molecule called grifixin. This is a broad spectrum antiviral. This grifixin is a natural product for the red alga Griffithsia. It produces a lot of grifixin from its leaves. So this grifixin we synthesize in the molecular biology lab. This is a 121 amino acid, like 12 kDa protein. It has a carbohydrate binding motif, mainly the mannose binding motif. So it effectively binds with the virus and on the envelope virus, not on the non-envelope virus, mainly on the envelope virus like HIV, HCV, MERS, and SARS coronavirus. When it binds to the virus with their envelope protein, the virus, it can't bind to the cell surface. So it prevents the entry of virus to the host cell. This grifixin molecule is a very stable, it is heat resistant, pH resistant, and it has no observed toxicity on the in vitro and in vivo for the host cells. So we are utilizing these molecules for the COVID-19 pandemic therapeutics. So earlier our lab has shown a beautiful result of this grifixin. We call it as QGRFT because this is modified from the GRFT. It has another amino acid modification. So our lab has shown this QGRFT therapeutics against SARS corona and MERS coronavirus. They are all SARS-CoV, MERS-CoV, SARS-CoV-2. They belong to the family coronavirus. So earlier they tested this molecule on MERS coronavirus and SARS coronavirus. When tested on the MERS coronavirus in 2016, they published the result in 2016 my lab. So they used the 3 cell lines, HUH7, MRC5, and Viro81. This is the control, non-infected. Other cells, they are the infected, but are treated with different doses of grifixin, 0, 0.125, 0.25, 0.51, and 2 microgram per ml. As you see, you can see a lot of virus, MERS coronavirus, that is red. It is the spike protein of MERS coronavirus. You see, without 0 means there is no grifixin. You can see a lot of virus in these cells, but as you see, like higher doses, this virus clears off. So this is the titration graph, how they titrated on the HUH7, MRC5, and Viro81.7, and with the increasing dose, they see a dose-dependent inhibition of the infected cells with around like 0.5 EC50 for the HUH7, 0.5 for the EC50 for the MRC5, and 0.25 or like 0.5 EC50 for the Viro81 cells. So they showed that this grifixin molecule is effective against MERS coronavirus. That is the same way, like it is the same as like SARS-CoV-2. MERS has the spike protein. So they predicted that this molecule, it can be effective against SARS coronavirus. So later on they tested the SARS coronavirus and they shown in vitro as well as in vivo. But MERS coronavirus, they have result for the in vitro. SARS coronavirus, they also show a dose-dependent decrease in the inhibition of the virus, and the cells are inhibited from the virus infection. Then they marked the mice with grifixin and concurrent treatment with SARS, and they observed that after the treatment, the animals were virus-free. They don't have any symptom for virus. They didn't die, and the mice, they were survived. So this treatment showed that grifixin work against SARS coronavirus. So now the papers have published from 5 years that is grifixin is effective against human coronavirus, human immunodeficiency virus, that is HIV, Nipah virus, and others. So it is like I will say this grifixin is a broad spectrum antiviral inhibitor. Now I will show you the SARS coronavirus result. These results are confidential. So this is our first experiment on the Vero cells, that is in vitro experiment on the Vero E6 cells. So we give the, we have the cells, Vero E6 cells, we give the virus as well as the different doses, started from 0.3 microgram per ml until 20 microgram per ml. We mix this protein with the virus so that the virus can bind to protein, and when we give it to the cells immediately, like mixing within minutes and then give the cell, it should not bind to the cell. But you can see like any treatment is not like 100% effective. So we see like very good efficacy at around like 5 and 10 microgram per ml. These are the cell control, these are virus control, and these are the treatment. As you can see, there is a lot of cytopathic effect on the in this Vero cell. They are only infected with the virus. You can see a lot of cells, they are dying because of the virus infection. But here in these 2.3 and 0.6 to 5, some of the cell, a lot of cell died in this 1.25 microgram per ml treatment. Some of the cell die, but here the increasing concentration, the death of the cell decrease in the dose dependent manner. So this is the graph that we generated from this data, like how much dose of beribicin we need to kill to protect our like 50% cell from the virus infection. So amount of effective concentration of beribicin come around like 1.31 microgram per ml, and this will protect like half of the cell from the virus infection. Now we expand this data to the human tissue. So we have this is like another experiment. So as we all know, the SARS-CoV-2 it infects the human cell with ACE2 receptor. So what we did in our lab, we have the Vero cells. So Vero has ACE2 positive as well as ACE2 negative. So we sorted the cell with the flow cytometry sorting machine, ACE2 positive Vero cells, and then we used these cells to treat the cell, to infect the cell, so that there like prevents infection of the cells and all of the cells should die within a few MI. So when we have this ACE2 positive Vero and virus infection, we propagate this virus in the ACE2 positive Vero cells, so that we should have a lot of PFU titer and we can titrate a lot of antiviral and other drugs on these ACE2 positive Vero cells. So this is our next regimen. So this is a cycle. So first we have to use the in vitro and then in between model and then animal model. So this is the in between model, the human 3-dimensional tissue. We use the epi airway and epinasal tissue for the beribicin prevention efficacy. So we treated the, so we have the epi airway and epinasal tissue. We treated the concurrent treatment, like combined treatment with the virus as well as the beribicin. We mix both of them and then we give to the epi airway and the epinasal tissue. So this is the cell control. You can see there is nothing virus titer in the, but in the MO.1 MI there is like 10 to the power 8. So this is, so we give the virus and then we collected the supernatant at different hour to see the virus progression and our progression, how we are preventing with the GRFT. So these are like, you can see at 120 hour, you can see at 0.1 MI there is a lot of virus, 10 to the power 8 CTCRD 50 per ml in the virus control, but in the, this is our beribicin treatment issue. So we have chosen different dose like 67 microgram per ml, 6.7, 0.67, 0.067 ml beribicin. So as you can see, as compared to control of any at hour, like we can at 72 or 96, 72 or 96 or 120 hour in the control tissue, there is around like 10 to 8 or 10 to 6. CID, but in our treatment there is around like 10 to the power 2 or 10 to the power 3 CID 50 per ml. We see a very good result with the epi airway with beribicin treatment, and then similarly we have the result with the epi airway tissue. So they are with the same manner, and but the efficacy was not good in epi airway tissue. So we have this efficacy, very good efficacy in epi airway tissue. Upon at the end of experiment, we need to see what is the mechanism behind this beribicin protection against SARS-CoV-2 infection. So we did the whole RNA sequencing of this their tissue, and we observed that. So this is the RNA sequencing data of SARS-CoV-2 infected and GRFT-treated human APRV tissue, because we see a lot of good virus inhibition in the APRV tissue. So, so when we compare the Griffithsin versus virus control, we see a lot of chemokine and cytokine. A lot of cytokine, they are membrane associated, like chitinase like 3, paralipin 2, platelet growth factor. But we see some of the cytokines that can be immunoregulator, like IL-33, DEPP1, ST3, GL4. Looking at the global picture, we see, I can say that there are a lot of cellular metabolic pathways that are involved with Griffithsin, but there are two pathways, that is hypoxia-inducible factor 1 and IL-17 signaling pathway. So they are playing them role. So in the immune response, there are three types of response. I will say now three types of response: TH1 response, that is the pro-inflammatory response; TH2 response, anti-inflammatory response; and TH17 response, that is the IL-17. This is a balanced response between TH1 and TH2. This TH17, it regulates TH1 as well as TH2 depending on the condition. So like SARS-CoV, in the SARS-CoV infected patient, there is a lot of pro-inflammatory cytokines like interferon, TNF-alpha. So if this GRFT induced TH17 signaling pathway, then it will go to the system and then in the immune system, and then it will regulate the TH2 pathway, so that there should not a cytokine shown as we see in the COVID-19 patient. So this is the very good data for the experiment that this GRFT is effective against SARS-CoV-2 infection. Now we are our next target. The leftover is the test this Griffithsin molecule against MERS coronavirus infection. And we have like, as you all know, this we don't have any model, well-established model for the mice model for the COVID-19. We are establishing ferret, hamster, as well as AC2 positive mice. That is they are AC2 mice, AC2 positive mice. They are humanized. They are transfected with humanized AC2 receptor, so that they should be susceptible to the COVID-19. So our next target will be to utilize the efficacy of Griffithsin against is in SARS-CoV-2 infection in AC2 mice, ferret, and hamster, and we have a good data coming for AC2 mice. So with all this, I will say thank you, Alpna madam, thank you, Dr. Pratima Srivastava, for giving me an opportunity to present our work at the CTRBEPR and JDB College. Thank you very much. These are some of my publications from here, and for cancer, diabetes, and infectious disease. If you have any question, please let me know in comment section, or you can have my email ID and you can ask any question. So there is like a lot of research opportunity for the people that are interested in virus infection, and because now in United States a lot of people they are working on the virus infection, and there is a lot of funding coming from the Democrat that they ask for the work on the infectious disease. So now if you have the student, they have calibration and interest for the vaccine research, for antiviral, or any kind of virus research, they can come and they can work. Initially they have to just only they have to publish 2 or 3 good papers. After that they will get a very good postdoc position here, and then they can work in their field or thank you very much.
Thank you, sir, for your very nice deliberation. Now, the participants, if you have any query, you write your queries in the chat box, and we will connect with Batra sir, and you will get satisfied answers thereafter. Sir, I want to know one thing from you, from my side. In the present scenario, can you suggest us some measures we should take to protect ourselves from this COVID-19 or the corona?
You are asking like preventive measure, like physical preventive measure, or like medicinal?
Yeah, medicinal side.
Medicinal side, yeah. Physical, because physical everybody knows, use mask, keep distance from others, 6 feet distance. So like medicinal, I will say like this, I will say like replacing, I will talk in like natural manner, like replacing this as a natural product. So I will say use natural product as much as we can possible. There is a lot of Indian medicine that we can use for this, so prevent the infection.
So to you also suggest that what Indian kadas and things we are from plant resources we are using, like Pinospora and all.
Dr. Pratima a very good remedy as well. It's failed in the clinical trial. So now they back up the remdesivir. So the remdesivir was a great drug for the COVID-19, but it's better. There is now no in the US nobody should treat with remdesivir. So I will say use natural product or like whatever you know in the traditional culture or in the ancient culture, because everything lies in our Indian culture.
Participants, I again request you, if you have any queries, write in the chat box. Over to you, Alpna.
Very nice presentation, sir. Thank you, Dr. Batra, for giving us information on antiviral protein in present scenario of COVID-19. Now, Dr. Mamta Chaudhary will introduce Dr. Somna Datta, Professor, Botany Department and Dean, Faculty of Science, University of Rajasthan, Jaipur, India. Over to Dr. Mamta Chaudhary.
Good afternoon all, and a warm welcome to our speaker, Dr. Somna Datta. I am Dr. Mamta Chaudhary. It's my privilege and pleasure that I have been asked to welcome my esteemed teacher, Dr. Datta, who is prominent researcher with a great experience of teaching for the last 34 years in University of Rajasthan, which happened to my alma mater. Honestly speaking, Dr. Somna Datta has tremendous work with State Forest Department to implement guidelines CBD Rajasthan. She has been motivated students and faculty across Rajasthan to work for biodiversity mapping and conservation in the communities. Presently, she is Dean, Faculty of Science. She will give her deliberation on indigenous communities and plant resource conservation, the way forward. So over to you, ma'am, Somna Datta.
मैसेज करो सामने मैंने कौन से देखा था मैंने
Connected.
Soon Somna Datta madam will join us. Madam Somna Datta will soon join us.
Yes, I'm on, Pratima. There's some problem with my video, you know. Can you hear me?
Yeah, ma'am, you're audible. Actually, such problems are considered as natural calamity. And in this era, we are well prepared.
So what I'll do, you know, I'll join from another device. There you can see the connectivity is there, but the video is not working there.
Okay, ma'am, you just carry on in whatever mode you are convenient.
Okay. So, can I start now?
Start, please.
Okay, so good morning to all. And I'm very grateful to the principal and organizers of JDB Girls College, Kota, for this opportunity. So let me begin today. My topic is indigenous communities and plant resources conservation, the way forward. What I would like to say was, you know, a little bit about the communities who are working towards conservation all over the world and also in India. A little bit of history, the societies who are engaged in this kind of work, examples of conservation, and what we can do in India, the way forward globally, as well as what we can do in India. So that is the kind of trajectory my speech will take. So we have to understand, you know, we live in a world which harbors many native traditions, and in India we know this fact for sure. This has been through centuries, and one of the things, the Agenda 21 of the Rio Earth Summit 1992, we have clearly realized the importance of indigenous people there, their traditional practices in environmental management and sustainable progress. We have to understand that they have survived centuries in this land, in Mother Earth, through various climatic changes, events, calamities, from polar to the desert areas, tropical rainforests, seashores to equatorial habitats, everywhere these communities have survived. So today I will talk about the future of the vast indigenous knowledge of our planet, what are the challenges they are facing, and how we move forward in the coming decades. Of course, I will restrict my talk to the plant resources only. So let me give you a little brief about the history, you know, the explorers who were rediscovering communities and their plant conservation practices dates back centuries ago. So uses of plants are found in Babylon and Egypt in 1770 BC, 1550 BC respectively. In India, we have our traditions from 4000 to 1500 BC. In 1777 AD, the Greek surgeon Dioscorides, we always teach about him to the botanists, published Materia Medica with 600 Mediterranean plants. So that was a big compendium of, you know, what communities were using plant resources as medicines, important medicines. In 1542 in Germany, there was Fuchs who documented 400 plants in Germany and Austria. So worldwide, all over, these kind of practices were being documented over these years. 1942, Columbus discovered America, and also he discovered tobacco in Cuba. So documentation of plants have begun in the 19th century in a more detailed, sustained manner. Humboldt, Cook, Charles Darwin, Palmer were great plant collectors and documenting the uses of these plants. In Bosnia in 1896, traditional medicinal uses of plants were studied by Glueck. Then we have, you know, in 20th century, the Zuni plants were recorded. There was this Cherokee community, they were studied and their traditions were studied. Closer home in India, of course, it was the greater India then. We had Burma, Pakistan, Afghanistan, all of this Indian subcontinent. We had these great Ayurvedic traditions, which goes back to the Rig Vedas of 4500 to 1600 BC, long ago. We know about many plants used for treatment of diseases in animals and humans, and natives of Indian subcontinent we believe, have a vast repertoire of medicinal plants used even today. And even during this pandemic, which our previous speaker was so eloquently speaking about, and the wonderful research he has done there, we have realized that, it is the biodiversity loss which may cause future pandemics also. Now, giving a brief view about the history, let's talk about the societies, what the societies were doing. Who were the famous ethnobotanists, who were studying these societies? Dr. Schulter was in, northeast Amazonian people. He was recording their records. Dr. Roder was meeting communities like Quanama, Ovambos. Turner and Bell was in Coast Saltish, Indiana, Vancouver Islands. There the communities and their practices, traditional practices, were being recorded. Indian communities also have been studied. We have a plethora of ethnobotanists and plant scientists all over India, spread all over the country, from northeast to Rajasthan to Kashmir and Ladakh Valley to the southern territories and the coastal territories. So some of the areas where it has been studied, documented, are Mayurbhanj, Araku Valley, Santhal areas, Andaman Nicobar Islands, Nagar and Haveli, Kumaon. Bheels and Gracias of Rajasthan have been studied. Khasis and Garos in the Meghalaya state have also been studied. These are just a few. Of course, there are many more tribes, and there's a vast literature in scientific journals about talking about the practices, traditional practices of these many communities in India. So idea is that, we have realized that now that forests and natural resources are being preserved on religious grounds. And these practices are known as sacred groves or diorais. In Rajasthan, we call them orans, where individual species, animals, are conserved by communities, by local people, by natural people who are staying there since centuries, who have made those forests their home. We call them the forest people. First, we used to call them the tribal people. Now this tribal word is not used anymore. It is the indigenous community we talk of. So in Rajasthan, I have also visited some areas, and I've seen the Jaisalmer Oran also. The Alwar one, Alwar also has some villages, and as many as 40 sacred groves are present there, where communities are trying to save plant resources there. Many plants, prosopis, people, ficus species, gooseberries, acacias, mango, indigenous, medicinal plants, they are all being preserved along with ethno-veterinary plants there also. Entada species is one important, plant which is being conserved in Sitla Devi Grove in Pune. Some other wild curcuma, that is wild haldi, which is being preserved in Kolhapur groves, sacred groves. Then we have the wild piper. Piper is the longum, piper longum, in Tamyani village of Maharashtra. The sacred groves also are playing a big role in conserving plant resources. And in sacred groves, it is the indigenous communities who are staying there. And we have kept a record of these communities in India as well. So this is a little about the history and some of the communities who are doing this. Of course, a vast number of communities all over the world. There is it in Iran, Bosnia, Bolivia, South American communities, and everyone has now come forward, and they are coming and talking to the United Nations and saying that, you have to make laws for protection of our land where indigenous people are staying. So now let us move a little bit to the laws, how we are protecting our resources. And a little bit about the Forest Conservation Act, we know in 1980, and it was amended in 1988. This checked the diversion of, what the act said was that you can't divert the forest land to non-forest use. You have to take permission for this kind of work. Then we have the protected areas law also, which can be used to manage. The protected areas have to be used to manage environmental resources. It is area of land and sea dedicated to protection and maintenance of biodiversity, its natural and associated cultural resources, and managed through legal means. This was the IUCN 1994 charter. So you have to maintain the culture culinary, their food habits, their plant resource, medicinal, their health, what they're doing for health, those kind of things also. And then in 1989, we have a record of about 4,500 protected area. And this is only 3.2% of the Earth's land surface, which is very less. We need more area for green cover. So next is the Environment Protection Act, which came in 1986. Then we have the Biodiversity Act, which was enacted in 2002 and enforced in 2004. Internationally, we have ratified many treaties and many instruments whereby we have to conserve our natural resources. The CBD, the Man and Biosphere Program, CITES, the Protected Areas Program, Kyoto Protocol, Nagoya Protocol, UNFCCC, many kind of such instruments, which are actually giving us a lot of force and a lot of motivation to go for conservation in every part of the globe. Let me tell you what has happened latest in this year. When the pandemic is going on, we are unable to meet, but this is the year when we have finished with the decade of biodiversity. The International Decade of Biodiversity was from 2010 to 2020, and this 2020 year, we are finishing this year of biodiversity. So the UN summit was held on 30th September. It was convened by the UN General Assembly, and people realized that the decade has ended. Now we have to plan for the future. What do we do in the next decade, 2020? So as the decade on biodiversity comes to an end in 2020, a post-2020 global biodiversity framework is being worked out. Everyone is getting together and we are working out what we have to do in the next 10 years, 20 years, 30 years till 2050. So I call upon our students, our young teachers to get involved in such programs, to know about this 2020, the post-2020 global framework, and then we have to work accordingly when we have to work towards plant resource conservation. Those kind of projects we will have to get according to the framework. The international community will come up with certain guidelines and we will have to follow that. So maybe a next seminar sometime in January, February, after this the framework is over, then we can discuss about what needs to be done now for in India also in Rajasthan. So a call in this summit, a call was made for mobilizing youth, business, women, consumers, and indigenous people and local communities to work for, the aim was to work for, I quote, urgent action on biodiversity for sustainable development and to pledge the 2050 vision of living in harmony with nature. So our 2050 vision is living in harmony with nature. We have to find ways and means and find out the practices by which we can live in harmony with nature, not fighting with nature, not fighting with one another, not exploiting nature, not exploiting people. These kind of things we will have to think about. So now I come to the last part, that is the way forward. How do we move forward? What do we do now? We now know the laws, what we have to think of, what is the status of conservation in the world. So how do we move forward, and what other people have done in Mother Earth to conserve the resources. So modern societies have to play a role in conserving ecosystem. I hope you realize that. We have to look towards our indigenous practices also, to learn from them, to learn best practices from them. And then we have to learn from other communities also in other parts of the world. Courts can play a great role by enforcing environmental laws. So the court has to do one part to enforce that justice prevails all over whenever some environment accidents happen or environmental encroachments happen. Other way is to grant legal rights to non-humans. This is a big thing which is being debated now all over the world, that let us recognize the rights of nature, like Ecuador has done, Bolivia has done. What they have done, they have granted rights to nature, non-humans, plants and animals, to ecosystems, to rivers. In Grandtown in Pennsylvania, what they have done is they have recognized the rights of natural communities and ecosystems. Some examples are in Colombia, where in 2018, the Supreme Court there ordered government to protect the Amazon and the Atrato River. So the river has to be protected now, and Amazon is actually saying that, yes, the river has a right to survive, right to live, so we will protect it. Then in India, here also we had something. We have shown concern by declaring animals as legal entities with rights, duties, and this was done by the Uttarakhand court in India. Even Ganges and Yamuna rivers have legal rights, but all these rights are, being challenged in court. But at least a way forward has been found that you at least start giving the ecosystems a right, like human right you must have nature rights also. Bolivia has recognized law of rights of Mother Earth of 2010. So Bolivia has done wonderful work. They have really actually gone and tried to speak about Mother Nature. What has happened in New Zealand? There was a treaty in 2017 between the Maori tribes and the government. They recognized the Te Awa Tupua as legal person, individual and living whole, comprising the Wangau River from the mountain to the seas. So there also this right was given to the Wanganui River. It's a nature-based right. So this is also a way forward. In India also, let us start off by saying that there is a right to Jhalana forest to exist. There is a right for Ranthambore to exist, other forest protected areas to exist. The communities will have to come forward to fight for these kind of nature-based rights. So this is another way forward where we can move. So such examples are of course not enough, because conflicts are always happening between humans and rights of nature. And we know examples which happened in Bharatpur. There was a big shooting there because people had sent in their animals for grazing inside the Bharatpur World Sanctuary. So is it the humans more important or the bird conservation more important? That became a conflicting idea. So you have to fight these kind of things and come up with a solution, viable solution. So this is the one of the way forwards. Then India, what we can do is, what we have been doing is that indigenous knowledge about plants we have a lot of medicinal plant knowledge in our repertoire. Healthcare, we have our indigenous practices of healthcare, and then we have indigenous practices of cropping patterns. Look at the jhum cultivation which is practiced in so many societies, a very typical way of crop growing. Then you have the veterinary sciences, where veterinary medicine, indigenous veterinary medicine is given to the cattle and animals in the farmyards. So all these indigenous knowledge, what we have to do now is to document it. And these must have a legal protection. So what we have done that, we have the traditional knowledge repertoire by communities. We are now documenting them, and these have a legal protection. How do we reduce overexploitation of resources? This kind of thing we can learn from the traditional communities, by the indigenous communities. There is one fabulous sacred grove in Khasi area near Meghalaya. It is called Maulilong Sacred Forest Area. How the indigenous communities have preserved that area, the plants there, the ecosystem there, the rivers and the mountains have been conserved over there. So from there people are going there. They have even started a little tourism project there. So people are coming to know about it, and they are also going and learning from them what they have done. How has this whole village survived so long? So it is one of the places on my wish list also, to go to Maulilong. And it was in India, it was also considered, I think it still has, as the cleanest village of India, right? So these traditional practices can be studied by us. So that is how we reduce the overexploitation of resources. Otherwise, there will be a point of no return. All our jungles and forests will just disappear, and all the resources will disappear with all the massive tree cutting and tree felling which goes on if we don't do something about it. And we have that example of Amrita Devi, who started that Chipko movement. The Chipko movement is now followed all over the world. It has become a powerful non-violent instrument to save forests. So NBA, the National Biodiversity Authority in India, is responsible for implementing the Biodiversity Act for conservation, sustainable use of biodiversity, access and benefit sharing mechanisms in this ABS instrument. So it is the Biodiversity Act, Biodiversity Board, which is responsible for a lot of these activities happening in India. They play a key role in India to manage all the biodiversity of India. So benefit sharing mechanism with knowledge holder communities has to be worked out. How do the communities benefit from the natural resources? If they sold some products, some fruits, some roots to a big company and they made some pharmaceutical medicines out of it, tablets and syrups and formulations. So what does the community gain who has managed these resources very well? And over years, they are supplying to the pharmaceutical companies. So there should be a sharing mechanism between both. Out of the profits of the companies, the communities also get some kind of monetary benefit. So this kind of mechanism is now being worked in India all over. Next, we come to partnerships. How do we forge partnerships? Partnerships are being forged all over the world between communities, with companies, schools and colleges and universities. That is where our role comes in. We can also forge, in Kota, you can forge communities with the partnerships with communities in Bara district, Kota district, what work they are doing, document their work. So I suggest our young botany and zoology students, young teachers, faculties to go meet the communities and document their work. That is very important. In Rajasthan, it is upon us to document this kind of work. Jobner is a place near Jaipur. We are asking our students to do some documentation work there. They have wonderful agro practices there. Then a student of mine is working in Kotputli. He has some wonderful agro practices there, documenting the plants there, the practices there, how they are managing their natural resources. All that is being so little effort by everybody, by universities also, college teachers in project form, individually, as a PhD project, one can do. So this is the way forward. We have to come. In fact, I was so happy when this theme was given as a, as a seminar topic, that now we can mobilize. It is a way to mobilize all our young community to work for this goal, which we have to reach by 2030. We have to, the target is that 30% of our land should be conserved. And now it is only about 8 or 9%. So it is a long goal. It is a difficult goal, but it has to be achieved when everyone starts working. So partnerships have to be meetings with indigenous people, challenges, what they're facing, try to find out. Otherwise, what is happening, challenging, the indigenous people are also coming into the mainstream. They are becoming city people, urbanized. They're leaving their ecosystems, their habitats. So that is now left for exploitation by companies, by other big people who want to maybe do a housing project there. So suddenly all our forest area is gone and a housing project or a dam is being built there, or some kind of a factory has come up there. So why the indigenous people need to stay there and practice and earn good livelihoods through their communities, that has to be worked out. And let me tell you that recently members of, it is not only in India, everyone in the world is talking about these things, what I am talking to you here today. The European Parliament met in November 2020. In fact, the meeting was just last week. What they discussed, they said that we have to discuss strategy for 2030. What can the communities do? And they called many groups from all over the world. Groups from India had also gone. There is one group which went from Assam, Chad in Africa, Central Africa, Congo, Colombia, European nations, Iran. Many communities attended this European Parliament, and they talked about the way forward, how we can move, what we can do to conserve our natural resources, what we should do by 2030, what we should do by 2040, and then 2050. Those were the targets which were being discussed. and partnerships are being forged. Europe is helping other people. Then Americas are helping. The UN is giving funds. There is a global fund also for these kind of work. Money is being sent for these projects, and we are absolutely, motivated, and we have to be challenged to take up this kind of work. You just can't say that only indigenous people will do this work. If we know some technology, let us share it with those people and we can go ahead. So this was the theme of my work. Just lastly, I will just mention one word, that what we have done in India is we have said that there is a category of forests called deemed forests. We have the forests all over the country. We understand that forest they have a record in the revenue records and in the forest department records. But there are many areas in India which are not recorded in these forest and land revenue records. But they are forests. They are being managed by communities. Sacred groves, they are not listed in these formal records. So that is a huge problem. So now we have to list these informal forests also, and they are being called as deemed forests. So Supreme Court has also taken cognizance of this, and they have said, you identify these forests. Every state of India must identify such forests which don't have a record in the forest department records, and you let us know. So all the states are giving a list of all their forests, deemed forests, to the Supreme Court, and they are going to mark them as deemed forests. So the deemed forest thing is also being working. We are working on this now, and all the states are really actively working on it. So that is how the Alvar people got together, because they have about the 40, 45, 46 Devrais, Dev Banis. One of them we also visited, 2-3 of them, with wonderful water bodies. They have protected their agrobiodiversity, ethno-medicine plants, a lot of things. So there they have made that record and they have submitted to Supreme Court. So this kind of work is going on. So our lawyers, if any of them are sitting here, they can work with our environmentalists, biologists, and work for environmental concerns also, where the laws are, where we have loopholes in laws and all that, that also can be addressed. Last, before just signing off, I must mention that we consider ourselves modern, but we must not forget that all our fathers, forefathers, ancients, we all came from Mother Earth. We stayed in this Mother Earth and we have survived by their practices, good practices. So when modern societies learn from our indigenous practices only, then can we save our environment from degradation. They have shown us the way. So there is a way to survive. We can learn from them. It is not a rocket science, we just have to handhold them and, handshake with them and start off this kind of work. Understand how they have survived, and we can also survive further. We have to save our environment from degradation, and the way is that we have to give rights to nature. This will resolve such conflicts and humans will learn to live in harmony with their surroundings. So thank you all. Any questions if you have, I'll be very happy to answer.
बैठ जाओ ना, बैठ तो मैं घुमा देती हूँ, बैठ जाते, मैं इसको घुमा देता हूँ.
Thank you, Somna ma'am. I hope our students and participants have definitely been benefited by your deliberation. One query is from our students only, like what are the research areas where students can work in the field of environment, biodiversity, and ecology? Ma'am, please throw some light on these topics and motivate our students.
That's a wonderful question. Like I just made one line or two lines in between the speech, that if you're staying in Kota, I'm addressing the Kota students, maybe Jaipur students, if my students are listening to me, you pick up panchayat level areas, panchayat. We have a panchayat, we have a sarpanch everywhere, and talk to the sarpanch and start documenting the plant and insect and butterfly and bird genetic resources there. Just start documenting it, what is present there, photograph it, and make a record out of it. We call this the People's Biodiversity Register. And this register is being made by the State Forest Department. We have a State Biodiversity Board who is doing this work. So this work will be given to you by them. They will train you how to do it. But as botany students, if you're a master's in botany or a graduate in botany, you can do this. It is not so difficult. Only difficult you will find in naming of the plants, the taxonomical part. For that, the resources are there to help you. The BSI is there to help you. The herbaria and the universities are there to help you. The expert faculty is there to help you. So start recording. This is one work. For this work, you will also get paid. You can do it in a project mode. You can take help of your teachers and submit a project to the State Biodiversity Department. We are asking all our students, and in fact, they have started this work also. I was saying my one student has started work in Kotputli. There are some students who have started work in Sambar area. So you can also do that. So this is one area. The other thing is, if you collect medicinal plants from these areas, if you collect food plants from these areas, something which is unknown, something which is a different species or different variety, try to analyze that in the labs. Find out its food value, find out why is it important, why do they eat this kind of grain, why don't they eat wheat, what we eat in the city. There must be some important elements, important chemicals. So phytomedicine, phytochemicals, that part you can do. Another thing is, if you can tie up with law, people start working on the environmental laws, where in societies where you have indigenous communities, where the laws are being enforced. If the laws are not being enforced, report immediately to the thing. This kind of work can also be taken up by the arts student, by the sociology students and all that. Then, of course, studying societies is the subject of anthropology and sociology. There is nothing called botany, zoology, research, it is now all intermingled. Now you start with your sociology friends, get them together and start studying the societies there, their cultural values, their music, their dance, their prayers, their rituals, their customs, all that has to be documented. This kind of work needs to be done in Rajasthan. And I request all the Rajasthan students that you have to take up this work. Look, we can learn from Kerala, we can learn from Karnataka, where this work is going on in full speed. Students are going out into the fields and documenting this work. Even in the Northeast, the areas, indigenous community areas, Arunachal, Shillong in Assam, there also students are coming out and doing this kind of work. So in Rajasthan, our students have no excuse not to do this kind of work. So just go out in the field and start doing this. Remote sensing is something which you can learn. We are also now mapping, our areas by remote sensing. That also you can do. It's a very interesting topic. Remote sensing is taught by the, Dehradun, even in Kazri, in Jodhpur. Just go stay there for 1 or 2 months and learn those techniques and start mapping the biodiversity, mapping of animals, their what do we say, the migratory routes of birds, and this can be done by remote sensing. So remote sensing work you can do a lot of things. So this dialogue, this can be, madam, another topic for a webinar, the research areas. We can even discuss about research papers. You must read up some research papers. Get in touch with us. We'll help you more if you want it, but we want that you all should work. Kota, there should be a big force of students working in these areas.
Thank you, ma'am. Obviously, I think that our students will definitely be benefited, and they will carry on the Atmanirbhar Bharat wala jo concept Modi ji ka hai. So by the way of research, they can earn also. And it's rightly said that think globally and act locally. So we have to act at our local areas. So thank you, ma'am. And over to Dr. Alpna Johari.
Thank you, Dr. Somna Datta ma'am, for your valuable suggestions for PG students and research scholars. Very nice presentation, ma'am. And thank you, Dr. Mamta. Now, there is a technical session. We have oral presentations of about 10 speakers. Now, over to Dr. Poonam Jaiswal.
We can take 5 minutes break. We can take 5 minutes break.
You can take 5 minutes break. After that, there will be presentation.
And don't leave the conference room.
Please don't leave the conference room.
We'll be back in 5 minutes.
थोड़ा सा prepared हो जाए. बंद कर कहाँ जा रहे हो? कैमरा बंद करो जाओ तो बंद कर दीजिए ना बंद कर Welcome back. Ready? Welcome to technical session 3rd. First, I am welcoming Dr. Tirthesh Kumar Sharma. He is presently working as Associate Professor and Head in Department of Botany, Bipin Bihari College, Jhansi. Dr. Tirthesh has 20 years of teaching experience with 24 years of research experience. His areas of specialization is microbiology, ecology, and environment. He has completed 2 projects and is postdoctorate by CSIR. He has supervised 7 PhD scholars and 1 MPhil scholar. He is National Fellow, GRF, SRF, CSIR, RA, CSIR, and SARC Fellowship. He has 34 research papers and 6 books published to his credit and has presented 70 societies and fellow of FES and FAELS. I have welcomed Dr. Tirthesh Sharma to present his paper on biomass, a source of energy. Welcome, Dr. Sharma.
Thank you very much, ma'am. Kindly allow me to present my slides.
Okay, sir. Okay, sir, I'm doing it.
Because the screen is showing the host disabled participant screen sharing. So kindly let me allow so that I can share my slides.
Okay, sir. Now you are a co-host. You can share your screen.
Okay, thank you very much. Thank you very much. Good afternoon, everyone participating in International E-Conference being held at JDB Government Girls College, Kota. Thank you very much for my introduction. The talk I'm going to deliver today is biomass, a source of energy. We are aware that biomass is an active mass present inside living organism, or we can define it as the organic mass in living organism. Biomass is accumulated in the plants through photosynthesis, where carbon dioxide is fixed in the form of carbohydrate, and this carbohydrate, after assimilation and synthesis, converted to biomass. About more than 1,000 million tons of solid waste are generated annually only in India. Of this, 375 million tons comes from the agriculture sector, and from agriculture sector, the sources are fruit cultivation, crop plants, and wood industries. Coming over to agriculture and forestry wastes, that comes from harvesting and clearing industries from the wood processing mill residues, and next is pulp and paper residues. Besides there are certain waste, other category of biomass is waste biomass. It includes crop forestry residues, animal manures, food processing wastes, yard wastes, municipal solid wastes, sewage and industrial wastes, etc. There are certain other category of new biomasses, that is solar energy and carbon dioxide solar energy and carbon dioxide converted by photosynthesis to organic compounds, and conventionally these carbohydrates are harvested for food, feed,
sir, sorry
and
to interrupt
fiber
you.
construction.
Sir, please share screen.
Yeah. I am sharing screen, ma'am. Just a minute. Sorry, screen. क्या करूं? यहाँ से बाहर जाओ. PPT तो है. Return में जाओ.
फुल स्क्रीन करो यहाँ से. ये
शेयर स्क्रीन फिर से जाइए. Now it's visible. देखो आ रहा है.
Yes, sir, is it visible? Go to slideshow.
Okay. Then coming over to what are the main agricultural crops that that are the main sources of biomass are sugarcane, wheat, sunflower, cottonseed oil, soy, jatropha, palm oil, rice, canola, and corn. These are the important agricultural crops which are the source of biomass. Now the problem is how we can eradicate it. As far as eradication is concerned, we have traditional biomass burning process and composting. But this biomass burning, which is commonly called as parali burning in the state of Punjab, Haryana, and other NCR regions, these added a huge problem of smog, development of smog over the national capital of Delhi. This we can visualize it, where visibility is almost reduced to nil. And this smog not only causes a great human problem, other problems to traditional biomass eradication are pollution. And because of pollution, when the pollutants will be added to in the atmosphere, it will bring about climate change, and any climate change will ultimately reduce to resource depletion. And we are also aware that every year our population is increasing, and increase in population will lead to energy demand. For example, to an estimate, by 2050, our population will increase by 60%, and it will increase in energy demand by about more than 100% or about up to 160%. There are renewable sources, the energy sources that we are using nowadays are natural gas, nuclear, petroleum, coal, and renewable energy. We can see that among renewable energy sources, out of total energy, it has only 6% of ratio. And among 6%, biomass has 47% of its contribution. So a large work has to be done as far as biomass is concerned. So again, the biomass, it's a renewable sources coming from biological material such as plants, animals, microorganisms, and municipal wastes. Where it is present, see, this is, if we see the cell wall of plants, outer wall is lignin followed by cellulose and hemicellulose. These cellulose and hemicelluloses are the carbohydrate biomolecules and are the important sources of biomass, or you can say these biomolecules are actually our target molecules that have to be converted into energy. Now coming over to bioenergy types, these are biomass can be converted into biofuels or bioheat or bioelectricity. Biofuels are of two types. One is liquid. Among liquid, it may be methanol, ethanol, butanol, or biodiesel, and gases, it may be methane and hydrogen. As far as bioheat is concerned, it's a wood burning process. Uh, this bioheat production we are practicing since ages, and bioelectricity, the wood can be combusted to boiler, and from boiler the energy can be transferred to turbine for electricity production. And another newer concept is microbial fuel cells. These conversion processes, if they involves microbial organisms or biological conversions, then it is called as fermentation. Through fermentation, we can have methanol, ethanol, and butanol. We can digest our solid wastes anaerobically with the help of anaerobic bacteria into the methane. Then anaerobic respiration, we can through anaerobic respiration, we convert it into biobattery for electricity production. And the chemical conversion, we can produce biodiesel through transesterification, and through combustion, gasification, and pyrolysis, we can produce heat energy. See, this is the whole process, how we can move from biomass to bioenergy. As earlier I have said, that biomass, through photosynthesis, biomass is accumulated, and it can, through conversion processes, it can be converted biofuels to bioenergy. Then it may be present in the form of wet biomass. Through anaerobic fermentation, we can have methane as well as hydrogen, and through this hydrogen we can drive our cars and motors. Among solid waste biomass, we have examples of wood and straw. It can be gasified, it can be combusted, it can be pyrolysis and hydrolysis. If we gasify it, we have fuel gas. This fuel gas can be used in electricity production, and if we combust it, and the energy produced by after combustion, it can also be moved, channelized into turbines, and from turbines it can be used to electricity production. In pyrolysis, where the biomass is burned at high temperature in the absence of oxygen, then we develop a char-like substance that is called as pyrolytic oil. This pyrolytic oil can be used to produce heat through heating. There are certain crops which are sugar cane, like sugar cane and several cereals are sugar and star-rich plants. This plant directly can be hydrolyzed and sugar can be extracted, and this sugar through fermentation can be converted to ethanol and butanol. There are certain oil crops and algae like soybean, sunflower. We can crush it and refine it and have pure oils in the form of methyl esters, to which we call it as biodiesel, or we can write it into liquid biofuels, and we can use it in transport industries. This process is called as transesterification. So if we convert biomass in biotechnological process, then we have certain advantages of it. First is that biomass is available in many parts of the world. Second, contribution to the security of energy supplies. Third, the cost is very low. Fourth, this resource can be regenerated. And second, it can create the jobs, especially in rural areas. Next, several technologies can be, other technologies can be developed and upgraded. And next, it can help in carbon dioxide mitigation and other emission related reductions, and reducing the harmful effect over the environment. But this biomass has certain drawbacks. For example, it has very low energy content. Second, competition for the resource with food, feed, and material applications like particles, board, and paper. So everything will, one day will come where everything will be channelized into for biomass. Then higher investment costs for conversion of final energy into comparison of fossil alternatives. The fossil alternatives means like petrols, gases that we have today. Biomass conversion may have a high cost. Now coming over to application points. Ethanol and butanol used in gasoline engines, either at low blends up to 10%. We can use it in blending with our petroleum. Then biodiesel can be used both, can be used alone, or it can also be used with after blending with normal diesel. These are the process how we can produce bioethanol from cellulose. Cellulose are the lignocellulosic material which are present in our forest or in our crops, agricultural fields. It has to be pre-treated. Then pre-treated biomass can be used for enzyme production or can be used directly for ethanol production after hydrolysis. And then after fermentation, it can be used for ethanol, and after recovery from the fermenter, this ethanol can be used alone or in mixture with the petrol in transport industries. Butanol is like gasoline, but it can be preferred over gasoline because it can be transported through the pipelines, whereas ethanol cannot, and similarly the gasoline cannot be transported. Biodiesels, we can produce biodiesel from triglycerides. Triglycerides are have the glycerol backbone and 3 fatty acid tails. This is the biodiesel production. How we can go for biodiesel production? Methanol use kiya jata hai. Then we mix the catalyst. After catalytic mixing, we add raw oils. Through trans-esterification and neutralization, methanol is recovered, and after phase separation, gravity centrifuge, then we have after purification and several steps of washing, we have finally we got biodiesel. Then the biofuels, as we know that biomass includes solid wastes also. These solid wastes can be digested anaerobically, and we have production of methane and hydrogen. So this methane can be used directly, either by it can be converted to ethanol or can be combusted directly or channelized through pipelines to the household. And this hydrogen can be used in fuel cells for the production of electricity, or hydrogen-based engines are being tried by automobile industries for use of hydrogen gas in transport industry. Then bioheat. This bioheat, before we use biomass in the bioheat production, certain steps have to be followed. First, it has to be dried in the sun so that the extra moisture present in the biomass can be removed. This process is known as torrefaction. After torrefaction, the color gets blackened, and then we can use it in controlled conditions. And then it is called as briquettes. And these briquettes can be used by small scale heating systems, household that we have been used since long time. Bioelectricity applications. After burning this briquettes, we can have electricity. We can transfer this heat energy to the turbines, and from there the turbines, the energy can be transported for electricity production. And last, Next, we have microbial fuel cells or MFCs, where the microbial cell direct conversion of can convert biomass to electricity. This is how the microbial fuel cell works. Here we have a microbial cell where the biomass gets anaerobically digested and hydrogen is produced. This hydrogen is accepted by cathode and this electron is transported and energy can be produced. Bioelectrochemical devices. Bacteria, in this system, bacteria acts as biocatalysts and can convert the biomass fuel directly to electricity through oxidation reduction reaction. In this reaction, the electron acceptor is a graphite anode. This is called as redox chemistry. This is how the microbial fuel cells works in laboratory. This is a whole view of how this bacterial cell or microbial fuel cell works in the MFCs through electricity generation. These are some of my research scholars who have worked with me and awarded PhD degree. And these are students who are working with me for their various topics. And thank you very much. Thank you very much. Thank you very much,
Dr. Sharma. Participants, if you have any queries, please ask in your chat box.
I should welcome. Yeah.
Please write in your chat box.
Dr. Tirthilesh Kumar Sharma, sir, has presented very nicely. And I hope the purpose of our conference is being fulfilled and our researchers and students are definitely going to be benefited. Students, please come forward and ask any queries in the chat box.
अच्छे से बोला कि आपकी वो
Thank you, Dr. Tirthilesh Sharma, for your nice and informative talk. The talk was very much relevant to the present scenario of pollution and smog in major cities of India. Thank you so much. Now I invite Dr. Deepika Lodha from JNV University, Jodhpur, to present her work on exploration of potent nutraceuticals value and conservation of Monsonia, an important endemic medicinal plant of Northwest Rajasthan, India. Welcome, Dr. Deepika Lodha.
Thank you, ma'am. Thank you, ma'am, for inviting me. Hello, ma'am. Am I audible?
Yes, ma'am. Ma'am, please you keep your mobile horizontal. Yes, ma'am. Okay.
Okay.
Post now. कहाँ है ये दीपिका? देखो तो है कहाँ?
Yes, ma'am. Good afternoon. Hello.
You are audible.
Yes,
Yes, ma'am Deepika Lodha.
Yes.
You are now
Yes,
a co-host, and you can present your paper by screen share. You can share your screen. ये क्या है? Ma'am, keep your mobile horizontal. Deepika ma'am, is there any problem?
There is some technical problem. Deepika will connect to you. Till then, you make your efforts. Now, next speaker, Dasani.
Now I invite Dr. Sippi Dasani for her presentation. Dr. Sippi Dasani is presently working as Assistant Professor, Department of Botany, Bipin Bihari College, Jhansi. Her areas of specialization is microbiology and environment. She has teaching experience of 10 years and research experience of 10 years. She has 15 publications and 4 books to her credit. She is a member of 5 learned societies and fellow of Academy of Environment and Life Sciences. She has presented papers in 25 conferences and has given invited lectures in 5 conferences. I welcome Dr. Sippi Dasani to deliver her lecture on bioremediation, a tool to clean environment. Welcome, ma'am.
Thank you so much, ma'am. Myself, Sippi Dasani from Bipin Bihari College, Jhansi. So today I am here to present my topic, bioremediation. First of all, the word bioremediation, what is it? Actually, we can see that it is derived from the combination of 2 words, bio and remediation. So bio means the living things and remediation means to rectify or to correct anything. And why? What is the need of bioremediation today? Because we can see that bioremediation is actually a term which is used since ages. Even in our Vedas, we can see that we always say that our surroundings or environment, basically,
Ma'am, sorry to interrupt you. Ma'am, sorry to.
It is made up of.
Ma'am, sorry to interrupt you. Ma'am, please
Yes, ma'am.
share your screen in slideshow.
Yeah.
Full screen slideshow.
Yeah. Is it number, ma'am?
अभी मैंने काट दिया, वो काके से बात कर दो।
Hello.
Ma'am, in right corner there is slideshow icon. Yes, ma'am. Okay, ma'am.
Yes. I have started. I think it might. Okay.
Okay, ma'am.
Yes. So coming to that, even in our Vedas, we have seen that Mother Nature or Earth, it is made up of Panch Tattva. That is Shiti, Jal, Pavak, Gagan or Samir, which in English we can say as earth, water, fire, sky, and air. And all those things in scientific terms are termed as mentioned here, we can see atmosphere, hydrosphere, lithosphere, which total overall contribute for ecosphere. But what we human beings are doing, actually, in previous lecture also I have heard that the human needs and the luxurious demands, we all go for maximum production. And these are the sources which are mentioned here, which are the main sources of pollution. That means the electronic gadgets we produce at maximum today. Anything we see the furniture in our house, even in the office which we are sitting, the production which we are going on doing without thinking about the mother nature is actually exploiting the environment.
वो गड़बड़ हो गया Zoom वाला, आवाज नहीं आई थी, ट्राई कर रही हूँ दोबारा।
To know that, we can see that the upper picture here is showing the mother nature in its most beautiful part, whereas the lower picture here is showing the more polluted side of the earth. What is our duty? Our duty is we have to maintain the mother nature as mentioned above. So for this thing, only the term bioremediation is applicable today, and it should be used in maximum amount today. So here you can see the definition. Remediation, as I have said before, it is to solve the problem to clean the environment, and with the help of living things. Again, coming to living things, they can be plants also, or they can be bacteria and fungi also, or even we can clean our environment. So if it is done through microorganisms, it is by nature, by natural means. Whereas if it is with the help of plants, it is the process which is known as phytoremediation. But human beings, if human beings will help to clean the environment, he will use the machines, and those techniques will be included under the conventional techniques. As mentioned here, we have mentioned very less conventional techniques. Just say here, that is biofiltration, pump and treat method, carbon adsorption, or landfilling. But these conventional techniques are actually very labor intensive and very costly and difficult to maintain, because it requires much effort as compared to the nature remediation. That is why, again, coming to why bioremediation, why not those conventional techniques if those are easy? So the main applications of bioremediation are, it is a natural process. Natural process means it is maintained by nature itself. We don't have to put any extra efforts for it. It is environmental friendly. Environmental friendly means when we go for conventional techniques, there are some toxics left behind in the nature again. But through bioremediation, we can excavate them. They are excavated directly and flooded during the nature. Then these bioremediational techniques are less expensive. These can be applicable in all the phases, that is solid, liquid, or gases. As these techniques have no side effects, they are socially accepted. Here are some microorganisms which are in nature itself and which actually degrade the pollutants. Some examples are cited here as Pseudomonas species, which degrades aliphatic and aromatic hydrocarbons. Then we can see halogens, which degrade species or halogenated hydrocarbons, and so on. There is a long list over here. Then coming to the types of bioremediation, actually it is divided into C2 and ex-C2 depending on the site where the remediation takes place. If the remediation is being occurring on the same site where the pollution has taken place, then it is known as in situ bioremediation. But once the pollutants are taken out from the original site, transported to the other site, and then they are irradiated, that technique is known as C2 bioremediation. So which one should be preferred? Again, there is no chance of preference because every technique has its own pros and cons. In situ methods are not costly or not labor intensive, but they are taking place only on the little space, and the experimental conditions are actually difficult to maintain at the same site where the pollution has occurred. And ex situ gives us better results, but it is very costly and very labor intensive to carry the pollutants from one place to another and then go for the degradation through the help of microorganisms. These are the points which are mentioned here. Same things are mentioned, that is, these are less expensive or they require much equipments. The other one requires less equipments and so on. Then coming to the forms of bioremediation, even whether it is an in situ or ex situ conditions, in situ conditions are cleared by bioventing. It can be biostimulation, sparging, bioaugmentation, or natural attenuation. What are all these? If we go for the term bioventing, it is the addition of extra oxygen or vacuum inside the soil or the layers of soil, which will actually activate the microorganisms which are detoxifying the pollutants at that site. In the same way, biostimulation means to stimulate the site which is polluted with the help of the things which we can provide externally to the microorganisms. Like suppose we say that bacteria is used to remediate the particular site, and that bacteria can survive at some particular pH or a particular amount of oxygen. If externally we can add that conditions to stimulate the bacteria, that process will be known as biostimulation. And same thing happens, suppose aspergillus is used to bioremediate the nature. There aspergillus survives only at 28 degrees Celsius, or it survives at the pH 4 to 5. What can we do? If we maintain the environment there, which is suitable for that fungi, we stimulate the conditions for the fungi to grow their maximum, which can detoxify the toxics there. In the same way, biosparging and bioaugmentation stands for the increment of the conditions. We can add the extra microorganisms there to the field or the polluted site, and we can add conditions also. Then coming to ex situ bioremediation. Ex situ means which can be carried out by land farming, composting, biopiles, bioreactors. For example, today it is carried out at maximum amount, that the kitchen waste or domestic waste, which is actually biodegradable. It can be carried from everybody's house or every kitchen to fields far apart from the cities where they are undergone for composting or land farmings. These are the techniques which are represented here diagrammatically, in which I have shown bioventing, biostimulation, bioaugmentation, and biosparging. These are the mechanical techniques, and the process of their stimulation or the anticipation of those chemicals or the conditions is mentioned here. Then here I have mentioned an example of genetically engineered microorganism, which is known as Pseudomonas for the bioremediation. So why we are going for genetically engineered microorganisms? Actually, it is not possible every time that maximum lot of microorganisms can be introduced in the environment where the bioremediation has to be taken place. So what a human being can do is mix the plasmid or the genetic composition of 2 or 3 or more microorganisms into and included into a single microorganism to get more improved quality of that microorganism, which can easily degrade 2 or 3 pollutants simultaneously. This is the main use to get the genetically engineered microorganisms, and here are few examples like Pseudomonas putida, which is used to degrade the xenobiotics like mono and dichloroaromatic compounds simultaneously. Then this is the method to produce the genetically engineered strain Pseudomonas. Here you can see xylene and naphthalene degrading plasmids which are present in 2 different bacteria, and below we have seen the hybridized variety of Pseudomonas putida, which is showing combined plasmid containing the sequence for both the pollutants to be degraded simultaneously. But in recent techniques, there is the best technique which is used for bioremediation today, and that is microbial consortium. What do we understand by this term consortium? Consortium is again a mixture of 2 or more microorganisms together, which will work for the detoxification of the pollutants at that site when they can be mixed either in different composition of microorganisms or different varieties of microorganisms. For example, only mono pollutants are to be degraded. Suppose one bacteria is used, but if we have to go for another degrant, then what we have to do is to add another bacteria. So mixture of both. Now we should know the proper ratio of both the bacteria to be mixed together. Sometimes the conditions may vary, sometimes the microorganisms ratio may vary. So we have to work a lot to develop various consortiums for the bioremediation, which is actually the demand of today. These are the proper actual sites of the bioremediation, in which we can see the first one. This is the air purification system. These are the sites of original solid waste biodegradation. And here, these are the pictures from Microbiological Consortium, Biodeg F company, which have shown many sites where the solid waste decomposition has been taking place with the help of microorganisms only directly in nature. Thank you so much. Thank you, ma'am.
शिवांगी तुम तैयार हो? शिवांगी से मेरा प्रेम हो गया।
Thank you very much, ma'am, Dr. Sipidasani, for your very relevant talk on bioremediation. You highlighted the need for cleaner environment using bioremediation in a very nice way. Thank you, ma'am.
Thank you, ma'am.
Now I invite Dr. Deepika Lodha from JNV University, Jodhpur to present her work. अभी नहीं आई।
Hello, ma'am. Hello.
मेरा टाइम पास यहाँ आप लोगों में कितना अच्छा हो रहा है।
हाँ मैडम। Welcome Deepika ma'am.
Thank you. Hello ma'am.
Now you are a co-host. You can screen share your work presentation.
Okay ma'am.
Yes. Ab she is ready.
Deepika, you're finding some problem?
Yes ma'am, I cannot share my screen, my presentation.
Okay.
Just wait ma'am.
In the meanwhile, Poonam, go for next speaker.
Paul, ready?
Deepika, you take your own time, no problem. After 2 presentations, you will be called. Next,
Okay, ma'am. Okay.
Now I invite Pahul Padam from Jalandhar to present the paper. Okay.
कृष्णा रानी मैडम के नीचे
एक मिनट
ये क्या है?
Good afternoon, everyone. Ma'am, I'm unable to share my screen.
Okay.
Yeah.
Paul, now you are a host, co-host. You can share your screen.
Yes, ma'am. Is it visible?
Yeah, Paul, you are visible, but not your screen.
Share screen.
Yes, started.
Okay.
Keep your mobile horizontal, Paul.
Okay, ma'am.
Okay. Yes, it's ready. It is ready now.
Okay. Good afternoon, everyone. My name is Pahul and I'm pursuing post-graduation in zoology from Kanya Mahavidyalaya, Jalandhar. And today it has been a keen privilege for me as I've been given this opportunity to be a part of this conference. Special thanks to my mentor, Dr. Smriti Johri, for providing me such an opportunity. I'll be presenting my presentation on evolutionary significance of digits in vertebrates. So, the area from where samples have been collected is Indian state of Punjab, sprawling in an area of 50,362 square kilometers. It comprises of 13 wildlife sanctuaries, 6 national parks, 4 conservation reserves, 3 community reserves, and 3 wetland sites. Now, the classification of vertebrates says that there are 5 classes of vertebrates: Pisces, Amphibia, Reptilia, Aves, and Mammalia. These are some of the characteristics. Next, the materials and methodologies involve a digital camera, clay mold, pen pencils, and it does not involve any harm to the animals. No pressure is involved and is safe and secure. Now I'm coming up with my samples collected through pictures and through the footprints on the molds. These are amphibians with webbed foot, with the mode of locomotion that is swimming, jumping, and walking. It belongs to order Enura, and it is these are the first tetrapods, and these are derived from pectoral fin of the ancestral fish. Next up is the class Reptilia, and it belongs to order Squamata. These are also tetrapods, and we have 2 examples here: Hemidactyla, that is wall lizard, and chameleon. These are tree climbers. Hemidactyla have sharp claws and adhesive discs. Here I'm talking about the footprints, digits and digits of various vertebrates on which evolutionary patterns, the changes in the evolutionary patterns are being involved. Next up is the tree climber. We have chameleon here. This chameleon digits are in opposable bundles of 2 to 3 digits. Another species is again from reptiles class, and it is turtle. It belongs to order Testudo. And the turtle is adapted with flippers and flappers. Next class is class aves. We have sparrow here and the pigeon. The claws of sparrow and the claws of pigeon are being taken. This is a sparrow passer domestica. The locomotion is hopping and the flight is direct. It is free running. If we talk about the pigeon, it is known as living airplane. They take up a quick jump without running. The next is peacock. This bird runs first. There are 3 digits present at the anterior position, and the third one is short and on the opposite side. Next we have duck. Duck is Anas domestica. It has webbed foot. There are 3 digits present in the anterior position. 1, 2, 3, and one is very short and is present on the opposite side. These are the footprints taken of this duck. Next we have, coming to mammals, we see here, this is rabbit, the squirrel, and cats and dogs. These are the footprints of rabbit. On the right side is the squirrel. So if we talk about rabbit, it belongs to the order Logomorph, and the locomotion is that is involved in the rabbit is crawling, dragging, hopping, walking, and running. It has elongated phalanx. Next, the squirrel. It belongs to order Rodentia. It has 10 pads under their paws. They have long claws to dig the ground. Plus, they can even turn their feet at 180 degrees without moving its leg. Next order is carnivora and involves cats and dogs. These are the pictures of, this is the footprint of cat and this is the footprint of dog. It possesses digitigrade foot posture, and the sole and ankle are raised from the ground. So there are 4 number of digits in both, and these are adapted for cursorial adaptation. Next we have ungulates. If we talk about ungulates, these are the grazing animals. We see they may be digitigrade and some bear single hoof only. For example, if we talk about this cow, buffalo, and goat, these are the examples of digitigrade, also known as dew claws. And it belongs to order Artiodactyla. And on the other hand, the animals with single hoof, they are hippos, pigs, and horse. I'm having the footprint of horse, and this SUS is the footprint of the pig. Next, in human, they are evolved with a platform with enlarged heel, and these are derived from from mammalia, and it has adapted to bipedal locomotion. So hallux, arc, hall, and ball of foot helps in the maintaining the balance of the body weight. Next, the comparison study leads to the corollary with the different species of different class, identifying their habitat adaptations, their evolutionary changes, and the literality level of the foot. These are the results, and the results says the species of class amphibia have webbed foot. If we compare the study, we have webbed foot of in class amphibia. The reptilia, they are pentadactyl digits with some of the bundled arrangement. In some hind limbs are adapted with paddles and flippers. Next class is the class apes, and these are the connected, and the collected link is Archaeopteryx. So their locomotion type is flying, but the pattern in the class aves is different in different species. As in the case of sparrow, it hops on the ground, pigeon make quick jump, and peafowl, these first runs and then jump, then flies up. We talk about duck, they have webbed feet. Now in class mammalia, we have funambulus. It has 10 adhesive pads. Rabbit have saltatory patterns. Cats and dogs, they have digitigrade. Digits are reduced to 4 now. Then the grazing animals, they have hooves and digitigrade, and single hooves are contained in some of the animals, in grazing animals. And the evolution from quadrupedal to bipedal locomotion is there in the case of humans. To wrap up, it says that this study helps in evolutionary studies, paleontological studies, and in various research fields. The change from quadrupedal locomotion to bipedal locomotion is the most advantage and is important to understand the foraging behavior and the distribution and patterns of animals. So these are some of the references. Thank you so much. I'm done, ma'am.
Thank you, Pahul.
As well as your mentor.
As well as your mentor, Dr. Smriti Johari. Thank you to both of you. Now I invite Pragya Dadij for her presentation. Pragya, you are now co-host. You can share your screen. Yes, agree.
Good afternoon, everyone. I am Pragya Dadij, a research scholar at Department of Botany, JDB Government Girls College, Kota. I am going to demonstrate environmental impact assessment on ecotourism in Ranthambore National Park, India. Pragya, आवाज नहीं आ रही. So, ecotourism. It is also known as green tourism. According to definition provided by the International Ecotourism Society, it is a responsible travel to natural areas that conserve the environment, sustain the well-being of local people, and involve interpretation and education, which leads to creation of knowledge and understanding. Ecotourism typically involves travel to destinations where flora, fauna, and cultural heritage are primary attractions. There are three pillars of ecotourism on which the entire concept of ecotourism is built. These are environmental stewardship. It involves initiatives and programs to protect and conserve the surrounding natural environment. Second one is sustainable business practices. It believes in minimizing enterprises' environmental impacts and maximizing its self-sufficiency, which includes everything from building material selection to water uses. Tourism and economy. Tourism is one of the booming industry in India and accounts for 6.5% of total GDP and about 6% of total employment. According to the report of the Travel and Tourism Competitiveness Report 2019, India is 34th out of 140 most visited countries in world. In economy of Rajasthan, which is one of the most visited states in India, tourism contributes about 15% of total economy and about 6.4% of state employment. Tourism is third largest employer in Rajasthan after agriculture and textile sector. Ranthambore National Park. It gets its name after Ranthambore Fort lying in the national park itself. It is one of the most popular parks for wildlife tourism in India and is famous for its large tiger population. It is 14 kilometers from Savai Madhopur district of Rajasthan. It lies at the junction of Aravalli and Vindhyan hill ranges. The present Ranthambore National Park was established in 1955 as Savai Madhopur Game Sanctuary over an area of 282 kilometers square. Over years, by relocation of 12 villages, inclusion of Kaila Devi Sanctuary and Savai Mansingh Wildlife Sanctuary, now it covers a total area of 1,334 square kilometers. It represents an ecological island surrounded by cropland and overgrazed landscape. Tourism in Ranthambore National Park. Ranthambore National Park consists of 10 tourism zones. Zone number 1 to 5 are known as premium zones, and zone number 6 to 10 are relatively new, opened in 2012 to meet the demand of tourism. Total area engaged in tourism activities is 13.14% of total area of national park, although NTCA permitted to engage 20% of area to tourism activities. There are no private vehicles allowed to enter in national park. Only gypsies and cantors can allowed in national park. There are limited number of vehicles, that is 140, can enter in national park at a time. Tourism remain closed in Safari Zone 1 to 5 during rains. Not only wildlife, but there are many things which attract tourists to Ranthambore. Some of these are shown in the picture. Plant diversity in Ranthambore National Park represents dry deciduous forest with about 539 species of angiosperm. Angiosperm pendula form the most dominant species and represented by 80% of flora. Profits of tourism, revenue generation. Ranthambore National Park is not only self-sufficient, but it generates surplus funds and increase foreign exchange. Ranthambore National Park became India's most earning wildlife park in financial year 16-17 with a revenue of 23.06 crores. The revenue generated by Ranthambore National Park is 2.5% of the funds provided by NTCA to the Ranthambore National Park. Ranthambore National Park provides employment opportunities for about 15,000 to 20,000 people, and and tourism improve value of land, infrastructure, facilities, and quality of life. The revenue generated for tourism contribute to preservation of historic properties, and tourism promotes social interaction between host communities and tourists. There are several negative impacts of tourism in Ranthambore National Park. These are, first one is environmental impacts, which involve pollution, inadequate disposal of solid waste. Most of the lodges and hotels in lying in Ranthambore National Park have no disposal plants, and they dump their waste in nearby villages or just burn them. Degradation of vegetation due to excessive tourism activities, soil degradation, and change in animal behavior. There was a tiger, T24, which became man eater and hunt many people. Then that was shifted to a zoo. Socio-cultural impacts involve resettlement, commodification, burden on infrastructure, urbanization, loss of local identity, value, and culture. It can be seen here that locals affected by tourist culture and tends to adopt that culture and forget their own culture and all. Economic impacts involve uneven economic development. It is seen here that the villages lying around the safari gates are much wealthier than villages which are lying distant from gates. Seasonal unemployment, high cost of land and housing, and other impacts involve introduction of non-native plant propagules, and pathogens. By ecotourism, why we should adopt ecotourism in place of conventional tourism? So conventional tourism have one goal only, that is profit. But ecotourism has three goals, that is profit, environment, and community. Conventional tourism is neutral to the ethics of environment and society, whereas ecotourism respect environment, culture, and social values. Conventional ecotourism focus on entertainment of tourists, whereas ecotourism focus on educational experiences. Conventional ecotourism in conventional tourism, conservation is not a priority, whereas in ecotourism, conservation of natural resources is a priority. The parameters that could be used for promoting ecotourism are reduced impact on environment, sustainability, interaction with nature, community involvement and interaction with people, environmental activities, economic sustainability. There are some measures which we should adopt in order to sustain the environment. These are, first one is eco-friendly resorts. Eco-friendly resorts adopt some key features which are recycling products, implementation of water and energy saving devices, proper disposal of waste, and conservation and expansion of forest. Second one is involvement of locals in consultations. No one knows an area better than its inhabitants. Local people are rich in traditional knowledge and can help policymakers for effective conservation practices. Ecotourism business by locals. Fourth one is consistent source of income, interaction between locals and travelers. It will benefit both locals and travelers. Involvement of local in plantation drive. During off season, all the locals which are involved in tourism industry should participate in plantation drive. Thank you. In the last, I would like to thank Dr. Poonam Jaiswal, my supervisor, and entire Department of Botany, JDB Government Girls College, for providing such a keen opportunity, for letting me present my ideas, and all of you for your time. Thank you so much.
Thank you, Pragya, for a very nice presentation. Now I invite Dr. Payal Sohn for his lecture. Dr. Payal Sohn is presently working as Assistant Professor in St. Wilfrid College for Girls, Jaipur. Her area of research is cytomorphology and biochemistry. She has teaching experience of 5 years and research experience of 10 years. She has 16 paper publications in various journals and 4 books published to her credit. I welcome Dr. Payal Sohn to deliver her lecture on anti-fertility effects of Calotropis persevera. Welcome, Dr. Sohn.
Thank you, ma'am. Good morning.
You are a co-host. All of you. You are a co-host. You can share your screen.
Okay, thank you. Thank you very much, ma'am. Presently, I am doing a small lecture on the methodologies which you can follow for your research work. Actually, whenever we meet some students, precious students who want to make their career in the research, they always ask in which field we can go for the research and how we can apply all the practical works and what will be the beneficial aspects of this research. So presently my presentation is on the work on which methodologies which we can follow for the research work, and we can use it in your research for a very fruitful and societal beneficial research. So I would like to present my screen. प्लीज 2 मिनट बस. Just a minute, ma'am. Is my screen is visible, ma'am?
No, ma'am.
Just a minute, ma'am. Actually,
Okay,
there is something.
Permission. Okay, ma'am. It's working.
It's working. You can see my screen. Okay, ma'am.
Ma'am, keep your mobile horizontal.
Yes, ma'am. Okay, the work is focused on the phytochemical and pharmacological methods which we can follow in the research work. So I would like to share the work how you will do in the research work. For that, we can use the medicinal plants. Actually, in our India and in our Rajasthan, there are a lot of plants which we can use for the medicinal purpose. And from the ancient times, it has been used for many remedies for the different diseases, cure, and etc. work. But now all the working industries on the medicinal purposes or the other purposes, we all now again focusing on these medicinal plants. So it is very beneficial for the new researchers who would like to do work in the research work on these medicinal plants. So my first focus is on the, firstly, you have to know about the what are the phytochemicals are present in that plant. Okay, so for that, you have to know which kind of phytochemicals are present in the plants. As you all know, there are two kinds of phytochemicals which are chiefly present in the plants. As we know, it is the primary metabolites and the secondary metabolites. So primary metabolites are those compounds which are present in the plant and which is used for its general purposes like the nutrition, growth, reproduction, and these are the proteins, lipids, carbohydrates, nucleic acid, etc. And the secondary metabolites are maybe the byproducts or maybe the intermediates of the primary reactions which the plant follow day by day. So these may be the flavonoids, alkaloids, terpenoids, phytosterols. So it can be secreted outside of the body, outside of the plant cells, or maybe in the cells. So these secondary metabolites are the key point or the key medicinal value purposes chemicals which we can be evaluated from the plants. So firstly, whenever we are focusing on the research work or particularly we want to go for the research, and the first thing we have to focus the particular plant which we are selecting having which kind of phytochemical. It may be flavonoid rich plant, or maybe phytosterols are rich in this particular plant, or maybe the terpenoids are there, triterpenoids are there. Like for example, Azadirachta indicana, the neem which we commonly know, it is very much rich in the triterpenoids, which has potential antimicrobial activity. So for this purpose, we only focus on the triterpenoids, we will extract it, or maybe for the other purposes like the alkaloids are rich plants, so we can go for the alkaloids, for the flavonoid which are very much rich, have a potential of antioxidant, so we can go for the antioxidant activities. So firstly, you have to know which kind of plant, which kind of chemical you are going for. So firstly, you have to focus on these type of plants, these type of metabolites. Further, I would do work on this plant कि मैं किसके ऊपर काम करने वाली हूँ, मुझे ये चीज पहले से पता होनी चाहिए. भाई आपको प्राइमरी मेटाबोलाइट से मतलब है कि आपको कोई पर्टिकुलर प्लांट से आपको प्रोटीन, जैसे कि हम बात करते हैं नॉर्मली कि अगर हम लेग्यूम्स वगैरह की बात करें तो हम फोकस करेंगे कि हमें फैट उसमें से चाहिए, या हमें चाहिए कि प्रोटीन रिच अगर हम बात करें तो वहां पे हम प्रोटीन्स की बात करेंगे. या अगर हम बात करें, जैसे आपने आजकल नए ट्रेंड में सुना होगा कि फ्लेवोनोइड रिच चाय है, तो वो फ्लेवोनोइड रिच चाय मतलब जो टी है, तो टी में ऐसे कौन से कंपोनेंट्स हैं जिसके फ्लेवोनोइड को बढ़ा रहे हैं, तो हम उसके बारे में बात करेंगे. ठीक है, तो वहां पे आपको पहले से पता होना चाहिए कि हमें किस चीज पे फोकस करना है, प्राइमरी पे करना है, सेकेंडरी पे करना है, सेकेंडरी पे भी हमें किस पे करना है. तो अगर हमें नहीं पता, हम रैंडमली अगर हम नया प्लांट ले रहे हैं, तो हम क्या करेंगे, सबसे पहले उसका पूरा फाइटोकेमिकल एनालिसिस करेंगे. For this phytochemical analysis, we will go for the qualitative analysis as well as the quantitative analysis. So qualitative analysis is based on the color, texture and appearance etc. So for that purpose, अगर हम जिससे बात कर रहे हैं, हम प्रोटीन की बात कर रहे हैं, हम अगर हमें किसी पर्टिकुलर प्लांट पार्ट में इसमें प्रोटीन निकालना है, तो हम क्या करेंगे, उसमें से हम उसको प्रोटीन को एक्सट्रैक्ट करने के बाद में उसके लिए हम बायोरेट टेस्ट कर सकते हैं कि हम उसमें हमें जो भी काम करना है। सबसे पहले देखिए एक चीज जो मैं क्लियरली करना चाहूंगी कि जब भी हम लोग फाइटोकेमिकल स्टडीज करते हैं तो हम ड्राई मटेरियल के ऊपर फाइटोकेमिकल स्टडी करते हैं, तो उसके अंदर हमें क्या करना होता है कि फाइटोकेमिकल जो स्टडीज होती है उसमें आपको हमेशा प्लांट को सुखा लेना है प्रॉपरली। पहले आप वॉश आउट करते हैं, क्लीन करते हैं, देन उसके बाद आप ड्राई करते हैं। ड्राई करने के बाद में उसको पाउडर फॉर्म में आप यूज करते हैं, एंड देन उसके बाद में आप इन सारे केमिकल जो आगे जाके आइडेंटिफिकेशन मेथड्स हैं, जो भी एनालिटिकल मेथड्स हैं, उनको आप फॉलो करेंगे। तो आपको सबसे पहले क्या करना है, उनको सुखा लेना है। ठीक है, हम ड्राई फॉर्म में इसलिए यूज करते हैं क्योंकि वेट फॉर्म में उसका जो भी फाइटोकॉन्स्टिट्यूट होगा उसका वेरिएबल आ जाएगा, जो भी, क्योंकि वहां पे फिर वो लाइव सेल्स रहेंगी तो वहां पे वो वेरी कर जाता है। तो हम ड्राई सेल्स को यूज करते हैं, उनको पाउडर फॉर्म में यूज करते हैं, एंड देन उसके बाद में हम उसको एक्सट्रैक्शन के लिए तैयार करते हैं। तो यहाँ पे जब भी अगर मैं बात कर रही हूँ क्वालिटेटिव एनालिसिस की बात कर रही हूँ या क्वांटिटेटिव एनालिसिस की बात कर रही हूँ, तो हम बात कर रहे हैं ड्राई पाउडर मटेरियल की। और ये जो भी रेफरेंस होता है, हम 1 ग्राम के अंदर हम सबसे पहले काउंटिंग करते हैं कि 1 ग्राम पर ग्राम के अंदर कितना है। ठीक है, तो क्वालिटेटिव में हम बात करें कि अगर वो उनके अपीयरेंस के बारे में बात करेंगे कि वो है कैसा, मतलब प्रेजेंट है या नहीं है। तो पहली चीज तो हम ये देखेंगे है या नहीं है, तो उसके प्रेजेंट के बहुत सारे टेस्ट होते हैं, लाइक जैसे मैंने बोला बायोरेट टेस्ट जो होता है वो प्रोटीन के लिए होता है, फैट्स के लिए आप आगे टेस्ट कर सकते हैं, लिपिड के लिए जो टेस्ट होते हैं वो आप फॉलो कर सकते हैं, आप उसका सॉलिबिलिटी टेस्ट होता है सबसे सिंपल, वो आप उसको फॉलो कर सकते हैं, स्टार्च के लिए आयोडीन टेस्ट यूज कर सकते हैं। तो इस तरह से अलग अलग टेस्ट होते हैं जो प्राइमरी और सेकेंडरी मेटाबोलाइट की प्रेजेंस को शो करते हैं। उसके अलावा हम क्या करते हैं कि हम जाते हैं इनको सॉल्वेंट के अंदर डिसॉल्व करने के लिए, जैसे कि जो अकॉर्डिंग टू देयर पोलैरिटी कि ऑर्गेनिक कंपाउंड्स कितने सॉल्वेंट में डिसॉल्व हो रहे थे, हम सिक्वेंशियल एक्सट्रैक्शन करते हैं। सिक्वेंशियल में हम क्या करते हैं, लोअर टू हायर चलते हैं। लाइक हम सबसे पहले पैटीथर में करेंगे, फिर हम उसके बाद में क्लोरोफॉर्म में करेंगे, फिर एसीटोन में करेंगे, फिर एथाइल एसीटेट में करेंगे, एंड देन फाइनली हम वाटर के अंदर हम उसको एक्सट्रैक्शन करते जाते हैं और उसकी क्वालिटी को हम चेक करते हैं कि भाई उसके अंदर उसकी अपीयरेंस कैसी आ रही है, कलर कैसा आ रहा है, उसका टेक्सचर कैसा आ रहा है, ग्रेनुलेटेड है, चिपचिपा है, ड्राई है, पाउडर फॉर्म में है। तो उसकी अपीयरेंस इन सबको हम नोट डाउन करते जाते हैं। तो आपको हर चीज का एक रिकॉर्ड रखना बहुत कंपलसरी होता है जब भी आप फाइटोकेमिकल्स के साथ में डील कर रहे हैं। देन हम बात कर रहे हैं उसके क्वांटिटेटिव एनालिसिस की, अब उसमें पर्टिकुलर अमाउंट कितना है। अगर हम प्राइमरी मेटाबोलाइट्स को चेक कर रहे हैं या सेकेंडरी को चेक करें, तो हम जनरली हमें सबसे पहले ही पता होना चाहिए इसका अमाउंट कितना है। तो अमाउंट कितना है उसके लिए अलग अलग मेथड्स फॉलो किए जाते हैं। ठीक है, उसके लिए सबसे पहले जैसे कि आजकल जो सबसे बेस्ट मेथड है दैट इज द GCMS एनालिसिस। GCMS इज गैस क्रोमेटोग्राफी एंड मास स्पेक्ट्रोस्कोपी जो मेथड होता है, इसमें आपको दोनों ही चीजें मिल जाती हैं। लाइक यू कैन गो फॉर द इसमें सारी के सारी जो भी हमें प्रेजेंट होती है, कोई भी कंपाउंड अगर प्रेजेंट है तो हमें उसके पीक्स मिलती हैं। ये पीक्स के अकॉर्डिंग हमें पता चलता है कि इसके अंदर कोई पर्टिकुलर कौन सा कंपाउंड प्रेजेंट है और कितनी क्वांटिटी में प्रेजेंट है। ये दोनों ही चीजें हमें इसके द्वारा मिल जाती है। सेकेंडरी है HPLC, वो भी सिमिलर ही मेथड होता है। तो यहाँ पे क्रोमेटोग्राफी जो होती है आप यूज कर सकते हैं। देन IR NMR, ये पुराने मेथड्स से जो अब इतने ज्यादा फॉलो नहीं किए जाते, लेकिन ये भी अगर आपको पर्टिकुलर किसी कंपोनेंट की स्ट्रक्चर को पता करनी है कि पर्टिकुलर प्लांट में कोई फाइटोकॉन्स्टिट्यूट है जो नया है और हमें आइडेंटिफाई नहीं है, उसकी स्ट्रक्चर आइडेंटिफाई करने के लिए IR और NMR को यूज किया जाता है, जिसमें कि हम जितने उसके हाइड्रोजन बॉन्ड्स हैं हम उसके बारे में बात करेंगे, और NMR से हम पता चलेगा कि उसके अंदर जो कॉन्फ़िगरेशंस है कार्बन का और उसके साथ में कोई डबल बॉन्ड बन रहा है या सिंगल बॉन्ड बन रहा है, कैसी स्ट्रक्चर आ रही है। तो IR NMR के द्वारा हम सारी जो स्ट्रक्चर है उसको एनालाइज कर सकते हैं। तो ये बात हो गई कि हमें किस तरह से कोई जो केमिकल है उसके अंदर क्या निकल के आ रहा है, एग्जैक्टली कौन सा केमिकल है जो हमें आइडेंटिफाइड हो रहा है। और इसमें मैंने एक और चीज बताई कि गो फॉर द प्योर एक्सट्रैक्शन के लिए अगर हम आगे जो प्रैक्टिकल करना चाहते हैं, फाइटोकेमिकल्स आपने निकाल लिए, लेकिन निकालने के बाद में अगर आप एक्टिविटीज के ऊपर जा रहे हैं तो आपको कॉलम क्रोमेटोग्राफी को यूज करना चाहिए फॉर दैट प्योर कंपोनेंट एक्सट्रैक्शन। अगर आपको पता चल गया, आपके पास एक की पॉइंट है, फॉर एग्जांपल जैसे मैंने काम किया हुआ है कैलाट्रोपिस के ऊपर, तो उसके कैलाट्रोपिस के अंदर एक केमिकल कंपोनेंट होता है कैलाट्रोपाइन, इट्स अल्कोलॉइड है ना, स्पेसिफिक होता है जो सिर्फ कैलाट्रोपिस के अंदर ही मिलता है। तो अगर आप कैलाट्रोपाइन को एक्सट्रैक्ट करना चाह रहे हैं, आपको आइडेंटिफाइड हो गया, आपने इसके सारे टेस्ट लगा के देख लिए, हाँ, और फाइनली आपके पास स्ट्रक्चर भी आ गई है, आपने ये देखा कि प्लांट के रूट्स के अंदर काफी ज्यादा मात्रा में है कैलाट्रोपाइन और ये शायद बहुत अच्छी एक्टिविटी शो कर सकता है। फिर उसके लिए मुझे काफी सारा कैलाट्रोपाइन चाहिए होगा, और ये जो काफी सारा कैलाट्रोपाइन प्योर फॉर्म में आपको मिलेगा वो कॉलम क्रोमेटोग्राफी के द्वारा ही मिलता है। तो आप फिर कॉलम क्रोमेटोग्राफी लगाते जाइए एंड देन उसको एक्सट्रैक्ट करते जाइए। देन उसके बाद में हम, जब हमारे पास में प्योर कंपोनेंट्स आ गए किसी भी पर्टिकुलर प्लांट की, एक प्लांट लिया आपने, 2 प्लांट लिया, 3 प्लांट लिया, आपने उसके पूरी बायोकेमिकल प्रोफाइल तैयार कर ली, आपको पता है कि इसमें एक पर्टिकुलर ट्राइटर्पेनोइड बहुत एक्सट्रैक्शन बहुत अच्छी मात्रा में है, या हो सकता है इसके अंदर फ्लेवोनोइड जैसे एंथ्राक्विनोन फ्लेवोनोइड या एंथ्राक्विनोन या कोई भी सेकेंडरी मेटाबोलाइट बहुत ही अच्छी क्वांटिटी में है। तो अब आपका फोकस होना चाहिए कि ये अब क्या काम कर रहा है, क्या हम इससे बेनिफिट ले सकते हैं। जैसे कि मैंने कहा कि नीम के अंदर एजिटेरेक्टा इंडिकाना के अंदर ट्राइटर्पेनोइड्स बहुत अच्छे हैं और वो क्या कर रहे हैं, एंटी माइक्रोबियल एक्टिविटी शो कर रहे हैं। तो मैं अब आगे जाऊंगी एंटी माइक्रोबियल एक्टिविटी के लिए। अब एंटी माइक्रोबियल एक्टिविटी में भी 3 तरह की हम बात करते हैं, लाइक एंटी बैक्टीरियल एक्टिविटी, एंटी फंगल और एंटी वायरल एक्टिविटी के लिए। तो इसके लिए जो मेथड फॉलो किया जाता है आजकल, जो फॉलो किया जाता है दैट इज द वेल डिफ्यूजन मेथड। उसके अंदर आप सीधे वेल्स के अंदर आप उसको अप्लाई कर सकते हैं, बहुत फास्ट मेथड होता है। और साथ के साथ में आप इसमें क्या होता है, मोर देन 10 सैंपल्स, उससे भी ज्यादा, इवन द 96 वॉल सैंपल आ गया है, आप उसको भी यूज कर सकते हैं इस मेथड को फॉलो करने के लिए। ठीक है, तो आप एंटी माइक्रोबियल एक्टिविटी के लिए जा सकते हैं। एंटी फंगल एक्टिविटी सिमिलर ही होती है, इसमें आप फंगल की कोई भी पर्टिकुलर स्ट्रेन आप लेंगे, उसका कल्चर करेंगे और कल्चर करने के बाद में उसके अगेंस्ट में एक्टिविटी आप लगा के देखेंगे। एंटी वायरल एक्टिविटी जो होती है वो चिक एम्ब्रियो के अंदर डेवलप की जाती है, तो आप वहां पे देख सकते हैं। इसके लिए लैबोरेटरी स्पेशलिटी होना बहुत रिक्वायर्ड है, और अगर कहीं पर है और शो कर रहा है तो दैट इज मोर देन, मतलब गुड से भी बहुत अच्छा है। क्योंकि अगर आप अभी प्रेजेंट सिनेरियो में देखें, पूरा जो ऑल ओवर वर्ल्ड है वो फोकस कर रहा है कि कोविड-19 के अगेंस्ट में या कोरोना वायरस के अगेंस्ट में हमें कोई भी ऐसा एंटी वायरल प्रॉपर्टी वाला कोई कंपोनेंट मिल जाए जो कि इसमें यहाँ पर हमारे जो कोरोना वायरस है उसको रिमूव कर सके, हटा सके या उसको किल कर सके। तो दैट इज द, किसको क्या कहेंगे हम, सबसे बड़ा हमारे पास की मिल जाएगी कि हम इस इतनी बड़ी डिजीज को या इतनी बड़ी बीमारी को जिसको हम पैंडेमिक कह रहे हैं, हम उसके अगेंस्ट में काम कर सकते हैं। तो काफी सारी लैब्स इसमें काम कर रही हैं, काफी सारे रिसर्चर्स लगे हुए हैं जो कि एंटी वायरल प्रॉपर्टी ढूंढने लग रहे हैं कि किसी प्लांट में, किसी एक्सट्रैक्शन में, किसी पर्टिकुलर डिकॉक्शन में ऐसा मिल जाए कि हमें एंटी वायरल प्रॉपर्टी मिल जाए और हम उसको यूज कर सके एज ए रेमेडी फॉर दिस पर्टिकुलर वायरस। उसके बाद में है लार्विसीडियल एक्टिविटी, जैसे कि आपके बहुत सिंपल सी बात है, जो मच्छर होते हैं बहुत ज्यादा प्रॉब्लमेटिक होता है मच्छरों के साथ में, तो वहां पे आप क्या कर सकते हैं कि आप लार्विसीडियल एक्टिविटी को कर सकते हैं। उनके जो लार्वास होते हैं, अगर उसके अगेंस्ट में आप कोई पर्टिकुलर प्लांट का एक्सट्रैक्शन यूज कर रहे हैं और वो लार्वा को किल कर रहा है, तो वो बहुत ही ज्यादा हेल्पफुल हो सकता है आगे इनको कंट्रोल करने के लिए। सिमिलर विद द निमेटोसीडियल एक्टिविटी, निमेटोड्स के अगेंस्ट में आप कर सकते हैं। साइटोटॉक्सिसिटी में क्या होता है कि अगर जैसे हर एक सेल जो होती है उसके जो क्रोमोसोम्स होते हैं, अगर वो डैमेज हो जाए तो आपको पता होगा कि म्यूटेशन कॉज करते हैं। तो इट मे बी हेल्पफुल, मतलब पॉजिटिवली भी और नेगेटिवली दोनों ही वे में ये आपको हेल्पफुल भी हो सकता है और माइट भी उस पर्टिकुलर जो ऑर्गेनिज्म है उसके लिए नेगेटिव इंपैक्ट भी कर सकता है। तो ये की पॉइंट लेके आप किसी भी कंपोनेंट की साइटोटॉक्सिसिटी कर सकते हैं, मतलब आगे जाके आप उसको डिस्ट्रक्ट कर सकते हैं या उसमें म्यूटेशन कॉज कर सकते हैं। कि क्रोमोसोम लेवल के ऊपर आप बात करें तो हम साइटोटॉक्सिसिटी वर्क कर सकते हैं। ठीक है, उसके बाद है जीनोटॉक्सिसिटी। जीनोटॉक्सिसिटी जो होता है उसके अंदर भी क्रोमोसोम में जीन्स लेवल पर जब आप चेंजेस कर रहे हैं तो हम जीनोटॉक्सिसिटी, मतलब टॉक्सिसिटी का मतलब यहाँ पे उनको खत्म करना है, ये पर्टिकुलर जीन्स को एलिमिनेट करना होता है। तो ये जो फंक्शंस होते हैं, जीनोटॉक्सिसिटी वर्क हुआ, साइटोटॉक्सिसिटी वर्क हुआ, ये हम बात करते हैं एनिमल मॉडल्स के अंदर। तो आप अगर रिसर्च में जाना चाह रहे हैं, थोड़ा सा एडवांस लेवल पे काम करना चाह रहे हैं, तो आप एनिमल मॉडल्स पे भी आप ये काम कर सकते हैं। ठीक है, अगर क्लासिकल बॉटनी से आप लोग जुड़े हुए हैं, आपको लगता है कि मुझे क्लासिकल बॉटनी में बहुत इंटरेस्ट है, मैं प्लांट से बहुत ज्यादा बिलोंग करता हूँ, मैं उनको आसपास देख के मुझे लगता है कि मैं उन पर ज्यादा काम कर सकता हूँ, फोकस कर सकता हूँ, तो हम उसके ऊपर वहाँ पे क्रोमोसोमल स्टडीज इज द बेस्ट पार्ट, जिसमें हम, क्योंकि देखिए इन्वायरमेंट से, अब यहाँ पे जब आप स्टडी कर रहे हैं तो आपको इन्वायरमेंट से कोरिलेट करना पड़ेगा कि आपके आसपास का जो इन्वायरमेंट है, लाइक द पॉल्यूशन है, किसी पर्टिकुलर एरिया में क्लीन इन्वायरमेंट में किस तरह के क्रोमोसोम्स मिल रहे हैं, आफ्टरवर्ड्स कहीं पे कोई पॉल्यूटेड एरिया है तो वहाँ पे क्रोमोसोम में क्या क्या चेंजेस आ रहे हैं, उसके अंदर क्या फर्क आ रहा है, क्या आपको म्यूटेशंस दिखाई दे रहे हैं, उसमें बाइवैलेंट टेट्रावैलेंट की स्ट्रक्चर कैसी है, उसके अंदर आपस में क्रोमोसोम चिपक गए हैं, क्या हो रहा है, व्हाट्सएवर जो भी चीजें हो रही हैं, तो ये सारी चीजें आप कर सकते हैं। ठीक है, देन उसके बाद में है आपका एपिडर्मल स्टडीज। जैसा मैंने कहा कि हम एनवायरमेंट से कोरिलेट करके स्टडी करें तो इसमें हम बात कर सकते हैं स्ट्रोमेटा के ऊपर। बहुत ही इंपॉर्टेंट थिंग होती है, आपने कुछ अगर देखे हो जर्नल्स वगैरह में देखा हो तो स्ट्रोमेटा जो होते हैं, क्योंकि हर प्लांट के जो स्ट्रोमेटा होते हैं अगर आप एरिया टू एरिया जाएंगे तो वहां पे इसका डिस्पर्सल भी अलग मिलेगा, उनके स्ट्रक्चर में भी बड़े और छोटे हो जाते हैं, साइज में बहुत वेरिएशंस आ जाते हैं, और अकॉर्डिंग टू एनवायरमेंट एंड क्लाइमेटिक कंडीशन दे मे बी वेरी। तो वहां पे आप इफेक्ट देख सकते हैं स्ट्रोमेटा के ऊपर, किसी पॉल्यूटेड एरिया का जो एयर पॉल्यूशन है उसका क्या फर्क पड़ रहा है, किस तरीके से हम उस पर काम कर सकते हैं, वो आप कर सकते हैं। रिप्रोडक्टिव बायोलॉजी है, रिप्रोडक्टिव बायोलॉजी में देखिए दोनों तरह के काम होते हैं। जो आपने बात की सेकेंडरी मेटाबोलाइट की, तो जो सेकेंडरी मेटाबोलाइट्स हैं दे मे बी यूज फॉर द एनहांसमेंट ऑफ प्रोडक्टिविटी, मतलब रिप्रोडक्टिव ऑर्गन्स का जो एफिशिएंसी है उसको बढ़ाने का काम कर सकते हैं। माइट भी जैसे कि हम बात करते हैं सफेद मूसली की, जो बहुत ही कॉमनली पहले से यूज की जा रही है हमारे ट्रेडिशनल सिस्टम्स के अंदर, कि भाई वो क्या करती है, उससे आपकी जो वाइटलिटी होती है वो बढ़ जाती है, रिप्रोडक्शन क्षमता जो होती है वो बढ़ जाती है, तो वहां पे हम उसको यूज कर सकते हैं। ठीक है, एंटी रिप्रोडक्टिव एक्टिविटी भी कर सकते हैं, लाइक द फॉर द जनरेशन ऑफ कॉन्ट्रासेप्टिव्स, कि अगर आपको ऐसा है कि कुछ पर्टिकुलर प्लांट है, जैसे गुलतुर्रा जो प्लांट होता है, सिसलपिनिया पल्चरीमा जिसको बोलते हैं, उसके जो सीड्स होते हैं वो यूज किए जाते हैं फॉर द एंटी फर्टिलिटी एक्टिविटी के लिए, या फिर जो प्रेगनेंसी टर्मिनेशन है उसके लिए भी यूज किया जाता है, तो आप उसको यूज कर सकते हैं फॉर योर रिसर्च वर्क। एनाटॉमिकल स्टडी की जा सकती है, इकोलॉजिकल सर्वे किया जा सकता है किसी भी जोन का जैसे रामसर, अभी आपने देखा होगा रामसर कन्वेंशन जो है वो उनके ऊपर वेटलैंड एरियाज के ऊपर काफी काम चल रहा है, गवर्नमेंट काफी फंडिंग कर रही है, उस पर काम किया जा सकता है। आपके अगर हम जयपुर में ही बात करें, फॉर एग्जाम्पल हम द्रव्यवती नदी की बात करें, अब जिसपे वर्क कर रहा है, करोड़ों सरकार खर्च कर चुकी है, तो वहां पर इकोलॉजिकल क्या डिस्टर्बेंस हुए हैं, क्या उसमें आप चेंज कर सकते हैं। जैसे कहा जा रहा है कि इतना पैसा खर्च करने के बाद में भी वहां पे अभी तक क्लीनिंग नहीं हुई है, सफाई नहीं हुई है, पानी साफ नहीं हुआ है, नदी का फ्लो नहीं हो पा रहा है, तो वहां पे बॉटनिस्ट या एज ए साइंटिस्ट आप लोग जाके उस पर्टिकुलर एरिया का सर्वे कर सकते हैं, उसको क्लीन अप कर सकते हैं, और वहां पे आप गवर्नमेंट को सजेशंस भी दे सकते हैं। ठीक है, तो उसके अलावा बायोरेमिडिएशन, जैसे कि मैंने कहा कि पॉल्यूटेड एरिया है तो किसी पॉल्यूटेड एरिया को क्लीन अप करना, माइक्रोबियल क्लीन अप के थ्रू आप जितना रैपिडली कर सके, तो किसी जगह को साफ करने के लिए आप उसमें फंक्शन आगे काम कर सकते हैं। इथनोबॉटनिकल स्टडी, इथनोबॉटनिकल जैसे कि इथनोबॉटनिकल सर्वे तो होता ही है कि हमारे ट्रेडिशनल जो लोग हैं, जैसे ट्राइबल प्रजातियां बहुत ज्यादा है, गरासिया है राजस्थान में, मीना है, भील है, काफी सारी यहाँ पे प्रजातियां हैं, मतलब जो ट्राइब्स हैं वो रह रही हैं काफी सालों से, तो उनका खुद का एक बहुत बड़ा ज्ञान है, बहुत सारी नॉलेज वो लोग रखते हैं, वो जानते हैं कि भाई कौन से पर्टिकुलर प्लांट की पत्तियों को यूज करने से हमारे घाव भर जाएंगे, कौन से पर्टिकुलर जो फल हैं उसको खाने से हमारी वाइटलिटी बढ़ जाएगी, बहुत सारी चीजें।
Excuse me, just to interrupt you, please sum up your presentation because we have many speakers in our pipeline. Please.
Okay, ma'am. It's just the last slide. I will sum up within a minute. Okay, ma'am. So afterwards, advanced botany में जो research करना चाहते हैं, they can go for the plant tissue culture, molecular diversity, gene identification, priming, bioinformatics, genomics, proteomics, drug designing, etc. So you can go for the many branches of the botany and the other works for your research work. So finally, it will be the award. You will got the Nobel Prize representing our India, and I wish you all the best who would like to go for the research and want to make their career in the research. Have a nice and wonderful day to all of you. Thank you very much.
Thank you, Dr. Sohn, for your such a valuable presentation on phytochemical and pharmacological methods used in research. Your presentation will definitely help research students working in this field. Now I welcome Dr. Vinisha Singh from Sanskar Bharati College, Jaipur, working as principal, to present her presentation. Ma'am, keep your mobile horizontal. Okay, ma'am. It's okay.
Am I audible, ma'am?
Yes, ma'am. Open presentation.
Am I audible, ma'am?
Yes, you are audible.
Okay, ma'am. Okay. Good afternoon, everybody, and welcome to e-conference on current trends in research in biodiversity, environment, and plant resources. I am Dr. Vinisha Singh, Assistant Professor, Department of Public Administration at Sanskar Bharati PG College, Bagru. Today, I want to tell you about goals of sustainable development in public administration. The SDGs are a set of 17 goals for the world's future through 2030, backed up by a set of 169 detailed targets, negotiated over a 2-year period at the United Nations, agreed to by nearly all the world's nations on 25th September 2015. There are 17 goals. Number 1, end poverty in all its forms everywhere. Poverty is defined as income less than 1.25 USD per day. The targets for this goal include a world where the poor are not vulnerable to climate change and have equal right to economic resources. Economic growth must be inclusive to provide sustainable jobs and promote equality. This goal aims to eradicate extreme poverty by year 2030. Number 2, end hunger, achieve food security, and improve nutrition and promote sustainable agriculture. Many still suffer from hunger and malnutrition around the world, mostly women and children. The food and agriculture sector offers key solution for development and is center for hunger and poverty reduction. Targets for this goal include ending malnutrition, protecting small farmers, protecting genetic diversity of crops produced, and investing in research to make farming more productive. Number 3, ensure healthy lives and promote well-being for all at all ages. Ensuring healthy lives and promoting the well-being for all at all ages is essential to sustainable development. Goal includes calls for universal health coverage, increasing healthcare workforce, reducing illness and death by pollution, comprehensive agenda for tracking a wide range of global health challenges such as AIDS, malaria, tuberculosis, vaccinate your family to protect them and improve public health. Number 4, ensuring include and equitable quality education and promote lifelong learning opportunities for all. Obtaining a quality education is the foundation to improving people's lives and sustainable development. Targets for this goal include the need for access to university level education, vocational training, and entrepreneurial skill. Help children in your community to read. Number 5, achieve gender equality and empower all women and girls. Gender equality is not only a fundamental human right, but is necessary foundation for a peaceful, prosperous, and sustainable world. Targets for this goal include freedom from discrimination and violence, ensuring equal share of leadership opportunities, and responsibility for women as well as property ownership. Several references are made to national opportunities in interpreting this target, call out sexist language and behavior. Number 6, ensure available and sustainable management of water and sanitation for all. Clean, accessible water for all is an essential part of the world we want to live in. Basic water scarcity affects 40% of world population. Targets for this goal include programs that provide access to basic technologies like toilet or latrines, as well as was protecting ecosystem that provide water, avoid wasting water. Number 7, ensure access to affordable, reliable, sustainable, and modern energy for all. Energy is central to nearly every major challenge and opportunity. About 1.3 billion people globally have no access to electricity. Targets for this goal emphasize a conversation to renewable sources and a dramatic improvement in efficiency everywhere. Use only an energy efficient light bulbs or appliances. Number 8, promote sustained, include, and sustainable economic growth, full and productive employment, and decent work for all. Sustainable economic growth will require
societies to create the conditions that allow people to have quality jobs. About 75 million people between ages 15 and 24 are estimated to be unemployed and out of school globally. This goal targets closing the gap using innovation as well as separating growth from ecosystem degradation. Number 9, build resilient infrastructure, promote inclusive and sustainable industrialization, and foster innovation. Investment in infrastructure are crucial to achieving sustainable development. Targets for this goal include fostering a more innovative and environmentally sound approach to industrial development. Think of innovative new ways to repurpose old material. Number 10, reduce inequality within and among countries. The world is massively unequal. To reduce inequalities, policies should be universal in principle, paying attention to the needs of disadvantaged and marginalized populations. Targets for this goal include financial market regulation to make the playing field more equal, making migration for ordinary, safe, regular, and responsible. Number 11, make cities and human settlements inclusive, safe, resilient, and sustainable. There needs to be a future in which cities provide opportunity for all with access to basic service, energy, housing, transportation, and more. More than half the world population lives in city. Target for this goal include addressing issues like transportation, disaster preparedness, as well as preservation of the world's cultural and natural heritage. Number 12, ensure sustainable consumption and production patterns. This goal targets topics like reducing food wastage, corporation sustainability practice, and educating people on the impact of their lifestyle choice. Recycle paper, glass, plastic, and aluminium. Number 13, take urgent action to combat climate change and its impact. Climate change is a global changes that affect everyone everywhere. This goal has an asterisk as references to Paris Agreement of December 2015, which guides nations to jointly commit to limiting global warming to not more than 2 degrees Celsius. Educate young people on climate change to put them on a sustainable path early. Number 14, conserve and sustainably use the oceans, seas, and marine resources for sustainable development. Careful management of these essential global resources is a key feature of sustainable future. The world oceans are overfished, underprotected, and stressed from climate change and pollution. The target of this goal include research and learning to find out more about what lies beneath the ocean so as better save them. Avoid plastic bags to keep the ocean safe and clean. Number 15, protect, restore, and promote sustainable use to terrestrial ecosystem. Sustainably manage forest, combat destruction, halt, and remove land degradation, halt biodiversity loss. This goal targets hitting the threat to living ecosystem and biodiversity on land. Plant a tree and help protect the environment. Number 16, promote peaceful and inclusive societies for sustainable development. Assist to justice for all and building effective accountability institution at all levels. Targets for the goal include reduction of violence, end of torture, reduction of corruption, as well as creation of institutions and government that work for everyone. Use your right to elect the leaders in your country and local community. Strengthen the means of implementation and revitalize the global partnership for sustainable development. Every country and every sector has a role to play in achieving those SDGs. This goal targets ensuring that countries have what is needed and achieving the rest, the SDGs such as funds, capacity, and technologies. The goal emphasizes the need for partnership and collaboration. Thank you. Thank you, ma'am.
Thank you, Dr. Vinisha Singh, for a very nice presentation on multidisciplinary field. Now I invite Dr. Deepika Lodha from JNV University, Jodhpur, to present her work on medicinal plant Monsonia heliotropides. Welcome, ma'am.
Welcome. Thank you, ma'am. Just a bit.
Are you ready, madam?
Yes, yes. ये देख लिया उसने.
Deepika, go ahead.
Mobile
Hello, ma'am. Am I audible?
Yes, ma'am.
Yes. Thank you. Thank you for inviting me. My topic is exploration of potent nutraceutical value and conservation of Monsonia heliotropodes, which is an important endemic medicinal plant of Northwest Rajasthan. I'm Deepika Loda, Assistant Professor, Government College Degana, and my mentor is Professor N.S. Shekhawat, Jain and Vyas University, Jodhpur. All the species and system of the nature deserve respect regardless of their useful to the humanity, according to Conservation International Annual Report, March 2014. The Indian Thar Desert and neighboring Aravalli provide unique environment conditions for growth, development, and evolution of biological systems. The biological and ecological importance of plant of fragile ecosystem are great in era of global warming, climate change. Desertic plants, Thar Desert जो plants हैं, may be harboring genes and product for bioprospecting, and many of these plant species yield secondary metabolite, gum, dye, nutraceuticals, and numerous bioactive product. So basically, जो Thar Desert के plants हैं, उनमें काफी सारी secondary metabolite, gum, dye, nutraceutical value के products हैं, जिन्हें हम explore कर सकते हैं, और काफी सारे plant भी ऐसे हैं जो unexplored हैं, जिसमें से एक है Monsonia heliotropodes, जिसकी family है Ziraniaceae. The objective of my studies were to identify the importance and problem of selected plant species, to study the factor affecting in vitro propagation and develop or standardize the protocol for regeneration and conservation of the selected plant species, which is Monsonia. This picture shows the natural habitat of Monsonia heliotropodes, which is an endemic and threatened plant species. The collections, this is a collection site of Monsonia, which is near Gajne Lake, Bikaner, and we collected Monsonia from Gajne Lake during 2014 in month of October. This is a natural habitat of Monsonia, which is xerophytic, hairy, rosulate appearance, and herbaceous plant. This is a very close near to the ground, and the land are very calcareous. This picture shows germination of seeds of Monsonia heliotropodes between deer droppings. On this is circle shows the footprint of deer. This shows the germination takes place with the help of deer only. This is a natural protection of Monsonia heliotropodes near Gajne Lake between the dried twigs, and we collected this plant from the Gajne Lake and transplanted and tried to maintain in the greenhouse. This slide shows the morphology of this Monsonia heliotropodes. This is an umbellate flower presented with long scap, and this is a flower in densely clothed with hair. Flower and whole plant is very densely clothed with hair, and in Hindi, Monsonia known as Mayur Shika and called as Mayur Shika because leaves look like fan-shaped crest or kalangi on the head of the peacock. Mayur Shika or Monsonia have a many importance. First of all, it is a source of antioxidant, have an appreciable amount of minerals, tannins, and proteins. It is used in neutralizing venom of snake bite. Monsonia leaves are directly chewed and used in epilepsy and mental disorders. And it is a favored food of deer. Why we selected this species? Because its growth is very habitat specific. It has low production, poor dispersal and poor germination of seeds, and it easily grazed and by the many animals, and so due to anthropogenic activity is become threatened. Earlier work done on Monsonia Heliotropoidis, or we can say plant tissue culture, is done on by Ram K in 2009 using seeds, and in vitro morphogenesis or callus culture is recorded. I used leaf and flower as a plant for induction of the callus, but plant grows very close to the ground of the surface, and plant is very hairy, so contamination is easily comes. This contamination is neutralized by multi-step and combined surface sterilizing treatment with ethanol, bavistine, streptomycin, tetracycline, and mercury chloride. So this slide shows the callus culture of Monsonia Heliotropoidis. Explant is umbellate flower. This is a callus induction on Murashiki Skoog medium with 2,4-D and Na is a plant growth hormone. This is a callus proliferated on kinetin TDZ and Na medium, and this is a green proliferative callus. But callus is very recalcitrant and not further differentiate into the whole plant. This is a callus from the leaf explant induced on MS medium with NAA and 2,4-D, and this is a proliferative callus with Na, Ba, and kinetin with 1% glucose, maltose, and 3% sucrose. The callus obtained from leaf explant is also very recalcitrant and did not differentiate further. Conclusion: Monsonia Heliotropoidis in vitro morphogenesis using two different explant, that is leaf and flower, has been studied. However, complete plant-led regeneration could not be achieved. I acknowledged CSIR, Council of Scientific and Industrial Research, Government of India, and many thanks to my mentor and Janan Vyas University for facility of lab. Thank you very much. Thank you, ma'am.
Thank you, Dr. Deepika, for a very informative and nice presentation. Now I invite Omkar Joshi from Mumbai to join us for presentation. Omkar Joshi.
Good afternoon. Yes. Good afternoon, ma'am.
Good afternoon. So you are now a co-host. You can present your presentation by screen sharing. Omkar, is there any problem?
Yeah. Yes, ma'am. I am trying to share, but
no, you are a co-host. Now you can share.
Yes, ma'am. I'm trying. Yes, ma'am.
Omkar Joshi, are you finding some problem?
Yes, ma'am. Yes, ma'am. I can't. Actually, I'm unable to share. Just a second, ma'am. Just a second. Ma'am, is it visible now?
Not visible. Not visible. Omkar, you can present after 10 minutes.
Yes, ma'am. Okay, ma'am.
Now I invite Shivangini Pandey to present her work on screening of cellulosic fungi isolated from degrading leaf litter of Saccharum officinarum. Shivangini, welcome.
Thank you, ma'am.
Go to slideshow.
Good noon to everyone. Myself,
Okay.
Shivangini. Ma'am, slideshow. Myself, Shivangi Pandey, research scholar, presenting my research paper on the topic screening of cellulolytic fungi isolated from degrading leaf litter of Saccharum officinarum. For this, I am very thankful to my guide, Dr. T.K. Sharma, for providing me facilities and guidance at every step of my research. Now come to the objective of this research. The main objective of this research is isolation, identification, and screening of cellulolytic fungi from the degrading leaf litter of Saccharum officinarum, which is the rich source of lignocellulosic material. So, I would like to start with introduction. So, cellulose is the most important carbon source on Earth. It is a linear, unbranched homopolymer of glucose subunit linked with beta-1,4 glycosidic bond. And cellulases are the enzymes which degrade the cellulose by cleaving the glycosidic bond and convert glucose into small subunits. And these cellulases are produced by a number of microorganisms such as bacteria, fungi, actinomycetes, etc. But among all microbes, the fungi are main producer of celluloses enzyme. For this purpose, the sample, that is leaf litter of Saccharum officinarum, was collected from the agricultural field of sugarcane located at Sonagir area of the district Madhya Pradesh and filled in nylon mesh bags. And these bags were placed into the pits for the degradation process. After the degradation process, the bags were taken out from the pits and the isolation and identification of process have been done. For the isolation of microfungi, the serial dilution method were used, and for the identification of fungi, I were used lactophenol cotton glue. After that, after the identification of fungi, the screening of cellulolytic activity of the identified fungal strain have been done. For this process, the identified fungal strain were grown on Zipac dox agar media and incubated at 30 degrees Celsius for their growth. After the growth formation, the platyprates were treated with 0.1% Congo red dye and washed with 1 m NaCl for the observation of clear zone. If the clear zone is present, it means that particular fungal strain have cellulolytic activity. These are the result of the this work, and here first column shows the name of the fungal strain, and second here here positive sign indicate the presence of cellulolytic activity, while the negative sign shows there was no cellulolytic activity at that particular fungal strains. And these are the photographs of fungal strains which shows the positive cellulolytic activities, and their name as Aspergillus flavus, Aspergillus japonicus, Aspergillus fischeri, Aspergillus nigrulans, Aspergillus niger, Aspergillus unguis, Acrinonium, Alternaria, Keto. Penicillium decumbens, Penicillium digitatum, Penicillium hercui, Phoma, Trichoderma, Trichosporum, Rhodotorula. So the final conclusion of this work is out of 46 identified fungal strains from the leaf litter of Saccharum officinarum, only 28 species having cellulolytic activities, and these cellulolytic fungal strains can be utilized to increase the rate of decomposition process in many other lignocellulosic conversion process in different industries. Thank you. Thank you, ma'am.
Thank you, Shivangini, for your presentation, and congratulations for your nice work. Now I welcome Omkar Joshi from Mumbai to join us for presentation.
Shivangini from New Zealand.
Shivangini.
सही है, शुद्ध है.
Omkar Joshi, finding any problem?
Hello, ma'am.
Shivangini? Okay, अब आई. Yes, Omkar, are you ready?
Yes, ma'am. Yes, ma'am.
Okay, start. You are already a co-host.
Yes, ma'am. Ma'am, is it visible?
Yes, you are visible. Keep your mobile horizontal.
Yes, ma'am. Good afternoon, everyone. So today I am going to present my work, that is
Omkar, please keep your mobile horizontal. Slide share mode में ले
Omkar Joshi, any problem?
Hello, ma'am. Hello. Am I audible?
Yes, yes, you are audible. Start presenting.
Yes, ma'am. Is my presentation visible?
No, it's a black screen.
Is my presentation visible?
Yes, कर लें दो ना.
Hello.
Yes, you are audible. You are audible. Start screen sharing.
Yes, it's visible. Oh, it's not.
Yes, your mobile screen is visible.
Shared. 1 minute.
Yes, yes. Okay. It's working now. Keep your mobile horizontal.
Yes. Yes, ma'am. Yes, ma'am. Okay. My topic efficient management of
You have
household
clicked stop.
biomass. These
Omkar, you have clicked stop share. You have clicked stop share.
Okay.
Yes. Okay. Okay, it's working now.
Okay. Okay, these are the contents of my
No, no, no. You have stopped sharing. आपने क्लिक कर दिया है स्टॉप शेयर को.
Is it visible?
You are visible, but your screen is not sharing. You have clicked stop share. कहाँ है ना? Yes, rotate your screen. Rotate your screen.
Hello, ma'am.
Yes, Omkar, carry on.
Yes. Is my presentation visible or my or no?
No, your presentation is not visible.
Okay, ma'am. Visible now, ma'am.
No, no, no. It's not visible. एक मिनट बस 3 बजते ही
I think
Is it
Omkar
started?
audio presentation.
Yes, ma'am. Sure.
Start with your audio presentation, Omkar.
Yes, okay, ma'am. I can give audio presentation.
Start speaking and explaining the things.
Hello.
Yes, start.
Okay, ma'am. So, good afternoon.
Sorry,
Yes,
Omkar,
good
your
afternoon,
time
everyone.
is,
Myself, Omkar Joshi.
time is up now. And I'm very sorry. You get ready for
Okay, ma'am.
tomorrow. And then definitely we are going to call you tomorrow.
Okay.
I would like to thank
Okay, ma'am.
all of the participants who have made this technical session a success. And for the inaugural session, I would like to thank Dr. Anita Kothari, Principal, JDB Government Girls College, Kota, Dr. Sanjay Bhargav, Assistant Director, Kota Region, and a special thanks to both keynote speakers, Dr. Lalit Batra and Dr. Somna Datta. They have spared their valuable time and presented nice deliberations, which are definitely important and on important issues of environment, and especially Dr. Lalit Batra, who has connected us to the present scenario of COVID-19. My heartfelt thanks to all oral presenters who have highlighted their research work in a very presentable manner. We are looking forward to meet you tomorrow. Till then, goodbye. And one more thing I would like to repeat, that tomorrow's presentations, after you attend the tomorrow's session, then only you will be given your certificates. So we have till then, goodbye.